Thanks a lot. So I got some emails back. Some of the students were busy with their classes and labs and exams, so they couldn't join us, but thank you so much for being here. So what I'm gonna do is, I'm gonna take their names, and they're gonna tell us who they are, what program they belong to, their research supervisors under whom they did their research from last year. So these are 2024, 2025 scholars, and they're just gonna let us know about their title of research, what they did, so just so that we know what the information was all about. All right, for the ACA Ledford Scholars, Wally, you were?
Good morning, everybody. My name is Wally Rayur. I'm a sophomore here at the University of Pikeville, and my major is biomedical sciences. My research topic for the Ledford Scholar was determine the mechanism of estrogen-induced G protein in the coronary artery. I know it's a handful, but that was my topic. I would like to say a quick thanks to my faculty mentor, Dr. Han, who really helped me through the project and getting the scholarship itself. I'd also like to say thank you to UPIKE, CHICOM, and just everybody else who's helped me along the way. Thank you.
Thank you so much, Wally. Do we have Colton Hall? Colton Hall here? All right. Connor McGuire? Nope. Alexa Muha? Yes, please.
My name is Alexa Muha, and I conducted research under the mentorship of Dr. Jessica Slade, and I was a recipient of the Ledford Scholars Program. My project focused on detecting antibiotic-resistant bacteria in soil samples from rural areas in eastern Kentucky and southwest Virginia. Soil is an important but often overlooked reservoir for resistant genes, and studying these environmental strains helps us understand how antibiotic resistance spreads beyond hospital and clinical settings. This research is important as antibiotic resistance continues to be a public health challenge that we globally face. By examining resistant genes and how they persist in the environment, we can begin to develop more effective strategies for monitoring and prevention. If you have any questions, please feel free to let me know. Thank you.
Thank you, Alexa. And yeah, a quick reminder, so most of these students are also presenting their posters. They might also be giving an oral talk. So if you have any questions about their projects, please get in touch with them. Next up, we have James Price. I don't see James here. Eva Roach.
Hi, my name is Eva Roach, and my mentor was also Dr. Jessica Slade. So I did research with the Ledford Scholars Program with Alexa Muha, but I also got the King's Fellowship Award, and I continued the project. So my project was a pilot study that focused on the isolation, detection, and identification of antibiotic-resistant bacteria, and we specifically focused on the strains that are resistant to ciprofloxacin and tetracycline. The next step focused on identifying those colonies that were specifically resistant to ciprofloxacin. Antibiotic resistance is a global health issue, so by trying to identify the specific antibiotic-resistant genes, we can have the potential to find alternative treatments in the future. If you have any questions, my poster is number 40. Thank you.
Thank you, Eva. There are some of these students that I've seen in the past 1 or 2 years as well. So these are students who have been presenting regularly every year, which is very, very commendable. Ethan Stiltner. Nope, probably busy with the classes. And Erica Worth.
My name is Erica Worth, and I'm part of the Chemistry and Psychology program at UPIKE. I conducted research under the mentorship of Dr. Han, and I'm a participant of the Ledford Scholar Program. My research was conducted to determine the mechanism of how estrogen promotes the effects of bradykinin on endothelial nitric oxide synthase, or it's also called eNOS for short, which plays a key role in cardiovascular health. Specifically, I looked at how estrogen influences the phosphorylation of eNOS at SIR1177, a step necessary for oxide release. Using a porcine coronary arteries, I treated the tissue samples with different combinations of bradykinin, estrogen, and receptor blockers. These findings suggest a potential treatment to reduce the risk of cardiovascular diseases and strokes. If you have any other questions, please stop by my poster. Thank you.
Thank you so much, Erica. All right, so moving on to the next set of scholars. This is the College of Arts and Sciences undergrad Scholars Program, CUS Program 2024. Connor McGuire got this scholarship as well, but he is not here. Eva Roach. Yeah, you've already introduced, right? Yeah, okay. Thank you. Thank you so much. That was really awesome, Eva. And then Laken Tackett. Nope. All right. The third and last is the Keene Family Fellowship Scholars Program 2024. There's only one scholar for that, Oksana Vucetic. Perfect. Thank you so much. All right. Thank you so much, everyone. Thanks a lot. All right. So now I would like to call Dr. Lori Worth. She's our Executive Vice President and Chief Strategy Officer for some opening remarks. Thank you, Dr. Worth.
Good morning, everybody. I'm going to try it again. I know it's cloudy outside. Good morning, everybody. There we go. Like I said to one table this morning, it might be cloudy outside and a little cold, but there's a lot of sunshine, hard work, and excitement here this morning. Thank you so much for being here. It's really interesting to see the evolution of a program. I was just speaking to our guests this morning on what it means to go back 6 years ago when we didn't have an event like this to celebrate the scholarship of our faculty, staff, and our students, and 5 years later to see something that is turning out to be pretty spectacular and now part of the fabric of the institution. I'd like to recognize the group of faculty who helped organize our symposium. If you were one of those individuals, I think there are 3 or 4 of you, would you please stand to be recognized? Dr. Worth, Dr. Nabil, Kaiko, okay. Let's give these folks a round of applause and thank them for helping us. We really appreciate it. If you are a faculty member here in the room, I would like to have you stand and be recognized as well. UPIKE faculty, please stand. Thank you. It takes a great deal of commitment to not only support student scholarship and research, but teaching and learning in the classroom. I've taught for about 15 years in several different colleges and disciplines, and there's a great deal of commitment to inspire students to be able to conduct research. It goes beyond what we find here at the university. The process of discovery is so very important. It's important to Appalachia, our region. So many of your projects and research studies are for the betterment of our region, given the health disparities that we have. We also celebrate the undergraduate students who are conducting research. I think there are many universities across the country that don't have the ability to support, and we're very thankful to our osteopathic medical faculty, our optometry faculty, and our new Tanner College of Dental Medicine faculty for partnering up with our undergraduate faculty members. A special thank you to our speaker. I know that we'll have some time to be able to introduce you. But Dr. Risher, we are so thankful that you took time from your full-time job to be able to join us in Pikeville at the University of Pikeville today. Welcome and thank you. We're excited for all of our student speakers today. Thank you to our faculty and staff, and have a wonderful day. Appreciate you.
Good morning, everybody. It is my distinct pleasure to introduce to you our keynote speaker, Dr. Mary Louise Risher. Dr. Risher is an associate professor with the Joan C. Edwards School of Medicine at Marshall University, a research physiologist at the Huntington VA Medical Center, and an adjunct professor at Duke University Medical Center within the Department of Psychiatry and Behavioral Sciences. Dr. Risher is a trailblazer when it comes to studying how adolescent binge drinking influences brain function and contributes to the development of alcohol abuse disorder, work that is funded by a grant she received from the National Institute of Alcohol Abuse and Alcoholism, a division of the NIH. She is currently funded by the NIH, Veterans Affairs, and the National Science Foundation. An accomplished researcher, author, and presenter, Dr. Risher has not only contributed to our understanding of the effects of alcohol on adolescent brain maturation, but has demonstrated a deep commitment to fostering the curiosity at all levels. In 2024, she received the Joan C. Edwards School of Medicine Dean's Award for Excellence in Research. As a passionate educator, researcher, Dr. Risher inspires others, demonstrating how research can have a profound impact on both academic communities and society at large. We are honored to have her here today to share her expertise and insight with us. So please join me in welcoming to the stage Dr. Mary Louise Risher.
Hello, and thank you so much for the invitation and the wonderful introduction. I'm very excited to be here. You have a beautiful, I've never been here before, so Pikeville is a really beautiful place. I got to walk around a little last night, which was wonderful, when it was sunny, before the rain kicked in. But I didn't start my career looking at the role of alcohol or binge drinking. I actually started in a lab that was primarily focused on understanding cognition using animal models, primarily rats, and looking at the effects of low-level exposure to pesticides and how low-level exposure to these pesticides would affect cognition chronically across the lifespan. So after that period of time, I also looked at a lot of cell culture studies, and I really felt like, because I wanted to understand mechanism, I wanted to understand the behavior, I wanted to understand mechanism, but at the time, we didn't really have the technology to be able to combine those things in the way that we can today. So some of the work that I'm going to show you today using some of these fiber photometric approaches is, for me, kind of a dream come true. And at the beginning, when I started my career, we also didn't really know a lot about astrocytes. We thought that they were just these puffy little things that sat around in the brain and just filled space and did very, very little. And now we're beginning to understand that they have this pivotal role in pretty much everything the brain does. So I'm super excited to share this with you. So I guess I have to stand here. I'm a walker, so I'm gonna start by talking to you about some of the trends that we see in terms of human consumption, and then go down this rabbit hole of how we got to the astrocytes and how we discovered their importance in alcohol research. I'll talk a little about the neuronal astrocyte interactions in terms of both structural and functional changes that we see, and the behavioral implications of those, because at the end of the day, what we are really interested in is the translational aspect of all of this. And then talk a little about where we're going next. So in terms of humans, everyone knows that lots of people drink. Especially in the US, in Europe, in England, where I'm from, there is a lot of drinking that goes on. If you look at these numbers here, in 2022, alcohol sales just in the US for 1 year were $260 billion. So there are some people out there that are making a lot of money, but please stick to research. Don't go off and start selling alcohol. Now, the most recent data that we have about excessive drinking costs was from 2010 and was already at $249 billion. So you can imagine that the cost of excessive drinking probably at this point is much higher than the amount that we see in alcohol sales. And there are lots of different things that contribute to these costs in excessive drinking. Law enforcement, collisions, injuries, work productivity, and chronic healthcare costs as well. But it turns out that about 77% of these costs are associated with binge drinking or some kind of excessive drinking. And I'm going to come back to this in a couple of slides and the importance of this. Staying with this human perspective for a moment, it's estimated that 1 in 8 Americans have an alcohol use disorder. So, take a moment and look around. There are probably people in this room that actually have an alcohol use disorder. But, there are many, many reasons why this is, why people don't talk about it, and we can definitely go into that. But what's, I think, most striking is that less than 10% of the people with an alcohol use disorder within the last year actually sought treatment. And once again, many reasons for that too. Now we see in the press that there's a lot of discussion about substance use disorder or illicit drug use. Obviously that impacts Appalachia significantly. But if you look at the numbers, you'll see right up here that we have twice as many people that have an alcohol use disorder than that have some kind of substance use disorder. So this is really very prevalent within the US. I think what is probably also somewhat disturbing is that approximately 3% of the people in the US between the ages of 12 and 17 were diagnosed with an alcohol use disorder in the past year. Very young population. And if you remember back a couple of slides, I mentioned that 77% of the cost of the economic cost of alcohol use comes from binge drinking. So if you look at the bar graph behind me and just focus on the green bars, that's binge drinking across the lifespan. And you'll see that it peaks around the late teens, early 20s, and then starts to dissipate as people get older. So, we're talking about 77% of the cost of binge drinking, 77% of the economic costs being from binge drinking, and the majority of that is coming from this adolescent period of time. And when I say adolescence, that's not meant to be disrespectful in any way, but our rats, which is a model that we use, age very quickly. So it's very hard for us to differentiate between late teens and early 20s. So when I say adolescence, I'm actually talking about these probably mid-teens to mid-20s in this period of time. Interestingly, so I should back up a second and say binge drinking, if you're not aware, is considered 4 drinks for a female, 5 drinks for a male across a 2-hour period. And if any of you were like my family, when someone pours a glass of wine, it's not usually what you would consider a glass of wine. It's usually a glass of wine. So when you count your drinks, you're probably drinking a little more than you anticipate that you would report on this kind of survey. In addition to that, college-age male and females consume more alcohol per binge episode compared to others. So, our criteria is 4 to 5 drinks over a 2-hour period, but most of college-age males and females are drinking in excess of that, significant excess. One of the things that we also need to keep in mind, and is very important for the work that we're doing, is that the earlier this drinking, heavy excessive drinking or binge drinking occurs, the more likely that person is to develop an alcohol use disorder later in life. So this is kind of the focus of a lot of the work that we do. So why do adolescents drink so much? Well, it turns out their brains are actually very, very different from an adult brain. And I'm hoping that this will resonate with some people in this room, because adolescents are less sensitive to the sedating and motor impairing effects of alcohol. So they don't have these internal checks that say, hey, you're getting sleepy, maybe you've had too much to drink. Or they start stumbling and you're like, okay, I've had enough. Those intrinsic changes aren't really happening appropriately in the adolescent brain because it's still maturing. However, adolescents are more sensitive to the rewarding properties of drugs and alcohol, including social facilitation, which, as a freshman, really is helpful, right? You're meeting all of these new people, so a drink may help to allow you to feel more relaxed and interact with these people that you're not so familiar with. They're also more sensitive to the memory impairing effects of alcohol, which means that when you do something rather silly or dangerous, you're less likely to remember it and repeat that behavior again. However, as an adolescent, you're out there partying and having a great time, but if you're around my age, you're probably still in bed sleeping by 9 p.m. The really amazing thing that excites me the most is that rats are exactly the same. Rat brain development mimics humans in all of these aspects. So when you go into the room and you have your rat colony and you have the adolescents and they see you coming with the alcohol, they get very excited. They get their alcohol and they have a little party in their cage. You do the same thing with the adults and they are literally just sleeping in the corner and they don't want to interact with anyone, which is just a beautiful thing from a researcher's perspective because this development really mimics the humans. So we know that this adolescent period is critical for the development of AUD, or contributes, I should say, to the development of AUD later in life. So I was part of the NADIA consortium funded by NIH earlier on in my career. And what we were really interested in was just understanding how binge drinking within that period of time would ultimately affect cognition, brain function in adulthood. And to understand whether exposure to alcohol during this period of time would affect the normal trajectory of brain development. So to look at this, we use an adolescent intermittent ethanol exposure paradigm that we refer to as AIE. So I might jump back and forth between binge alcohol and AIE a lot. The model that we use is, well, primarily at the beginning we were using male Sprague Dawley rats. We've now moved into male and females. But at postnatal day 30, so that's 30 days of life, we orally gavage animals with 5 grams per kilogram of alcohol, which brings us to about 170 to 190 mixed per deciliter. And to put that in perspective, 80 mg per deciliter is the driving limit. College students, depending on what they're up to, can reach about 250 mg per deciliter. At that point, you're kind of looking at hospitalization in many cases, but this 170 to 190 isn't really a blood alcohol level that is going to put them in the hospital, but they're going to be highly intoxicated. And when we see those characteristics in these animals. So we intermittently dose these animals with this binge level 10 times across 16 days. We then allow them to go through this washout period or period of abstinence until they reach early adulthood at postnatal 70 to 76. So some of the work from the consortium across all of the labs found that if you dose these animals in adolescence, allow them to grow into adulthood, what we see are chronic decreases in the ability to problem solve. Cognitive flexibility is impaired. So that's going to be prefrontal cortex areas. Information processing speed is decreased and impulsivity is increased. And you can see how maybe some of these things may contribute to bad decision making. So, you know, there's definitely a role for some of these permanent changes that may be occurring. Now, if we dose these animals in adolescence, allow them to grow into adulthood, and give them just one binge dose of alcohol, when they're acutely intoxicated, this is the interesting part. We see a decrease in the sedating and motor impairing effects of alcohol, and an increase in the rewarding effects, and the increase in the memory impairing effects of alcohol. And you'll see this looks just like our adolescence, right? So we're not sure. Oh, and when we do this in adults, the same paradigm, but start in adulthood, we don't see these changes at all, suggesting that this adolescent period is really, really critical for developing these cognitive changes that are likely going to increase consumption, but also in both ways, right? So we have a decrease in this cognitive flexibility and processing and more impulsivity, but those built-in checks that you should have as an adult aren't there. So they're also likely they're also likely to drink more as well. So we don't know whether this is an adolescent phenotype that is kind of locked in from adolescence and persists into adulthood, or whether this is just simply dysfunction that is reminiscent of adolescence. So we wanted to dig a little deeper. We looked at, so we decided to look at the circuit level. So here we're looking at the hippocampus, which is obviously critical for learning and memory, more learning than anything else. But what we're able to do is take these acute slices from these animals, place them in an electrophysiology rig to keep them nice and happy, and we can essentially stimulate these circuits in a way that replicates learning, and that we also refer to as long-term potentiation. And the idea is that what we actually found was that in these animals exposed to adolescent alcohol, it's easier to stimulate these neuronal circuits. And that is something that we typically see in younger animals, hence the reason that younger people learn more easily, and 10-year-olds like my daughter can learn their spelling list very, very quickly, and these things will become harder as we get older. So we dug even deeper and we said, okay, if these circuits are more plastic, then what is happening at the synapse? Is the synapse more plastic? So here we have a presynaptic terminal and a postsynaptic terminal, and the postsynaptic terminal is actually comprised of what we call a dendritic spine. And the morphology of these dendritic spines really influences the activity of these synapses. So you'll see that we have a mature, we have, I'm going to turn around here. You can see Philopodia and the long fin. They represent the more immature dendritic spines, which we consider to be the learning spines. They're very plastic. They can be removed very easily and replaced very easily. Whereas the stubby and more mushroom spines are the more mature spines that are very, very stable, and we consider those to be the memory spines. Those terms aren't specific, but it's probably the easiest way to kind of describe their roles in the brain. And what we find is that we see a shift towards this immature dendritic spine phenotype as well, suggesting that these synapses aren't maturing appropriately, and that may be driving the immaturity that we're seeing at the circuit level. However, these synapses don't reside in a bubble. Many of them are ensheathed by this peripheral astrocyte process, these processes coming off of these astrocytes. And these astrocytes play this really critical role in synaptic formation, maintenance, and homeostasis. So they're also really important for cross-communicating with the synapses and helping them mature appropriately. So, they also have lots of different roles as it's described here. They also interact with blood vessels or the vasculature of the brain via these end feet. So they allow nutrients to come in, waste to go out. They interact with these synapses. They interact with each other and have lots and lots of different functions that we're only just now beginning to really understand and recognize. But it turns out that in 2007, Kristen Harris's lab actually used EM reconstruction and demonstrated that these mature synapses are actually larger when astrocytes are present. So that made us wonder whether these astrocytes are actually not positioned structurally appropriately at the synapse to promote synapse maturation or dendritic spine maturation. So we went back to this model. We did the same thing again, this time using 2 time points, 24 hours after the 10th dose or the last dose, and then our typical adult time point. And we did immunohistochemistry and probed for this glial fibrillary acid protein, or GFAP, which is specific for astrocytes. It stains the backbone of the astrocyte. And when there's astrocyte reactivity, when the brain is unhealthy, when there are bad things happening there, we tend to get an increase in GFAP expression and an increase in branching. So that was our first indication that maybe something was happening with the astrocytes, that they weren't necessarily happy. And what we essentially found was that immediately after the alcohol exposure, there was no change in GFAP at all. But when we look after the abstinence period, what we see is an increase in the GFAP expression and the branching of GFAP. And in addition to that, what we're also seeing is an increase in astrocyte signaling factors that are secreted from the astrocytes. The problem with this is that these secreted astrocyte factors are only meant to be upregulated twice. Well, in 2 situations. Once is during development to promote synaptogenesis. The other one is when there's an injury. So for instance, a stroke. About 10 days after a stroke, you're going to see upregulation of these factors that are going to promote synaptogenesis and recover the function of that area. And then they're going to be upregulated transiently, and then they're going to decrease again. What we're seeing here after this alcohol exposure, only after this abstinence period, is a chronic upregulation of these factors that should not be there, suggesting dysregulation of these astrocytes. So this was the first step, the first indication that maybe astrocytes were playing an important role. But really, we still hadn't answered this question about whether astrocytes are physically appropriately ensheathing these synapses to provide the support that they need to provide. And at the time when we first did this experiment, the best thing that we had was this GFAP marker, which, as I mentioned, only stains the backbone. But it turns out we now have the technology to be able to use adeno-associated viruses that we can intracranially inject into the brain to the area of interest and target them to our cell of interest, which for us would be the astrocyte, and incorporate this green fluorescent protein in the membrane of these astrocytes. So you can see here that when you look at the astrocytes that were, wrong side, that were used for the immunohistochemistry, we're only seeing the backbone. But if you look at what we are able to achieve with this AAV, this is actually what the astrocyte looks like. Huge difference. And if we compare the rodent astrocyte morphology at this 3D level what we realize that they're much, much more complex than what you'll see in the fly or the C. elegans. However, when you compare them to a human astrocytes are 10 to 20 times the size of a rodent astrocyte and much more highly complex. In a great paper that was published by Macon Nedegard, actually they were able to take immunosuppressed mice. They were able to take these astrocyte progenitors, put them into the brains of these mice, and the astrocytes incorporated fully into the circuitry, and they were actually able to improve cognition in these mice, suggesting a really, really critical role for these astrocytes in cognition. So this actually comes from a human data set. Serial EM reconstruction was done on an entire human brain. And you can see that the complexity of this, right? If we know that a single human astrocyte can make contact with 1 to 2 million synapses, and we've only ever looked at neurons, we have this whole environment in which the brain is communicating that we haven't even figured out yet. You know, if you think about the neurons being your highways, and that's everything that we've always looked at. Everything runs by the highways. But if you think about these astrocytes interacting with each other, talking to each other through these alternate pathways, which you can think of as your secondary roads, you have a huge amount of connectivity and communication happening in the brain that we haven't even tapped into, even for a moment. And in some ways, as a neuroscientist, it's really, really overwhelming, but also exciting at the same time. So to give you an idea of what this looks like, we can actually do 3D reconstruction of these astrocytes in our rodent brain. So the green signal is the AAV targeting the membrane. The magenta is the GFAP, so the backbone of that astrocyte. And the red puncta is a synaptic marker. So what we can do is a surface rendering of this entire astrocyte and ask, is the surface of the astrocyte co-localizing within 0.5 microns of this synapse? And really start to understand how alcohol is influencing these interactions. So this work actually started with one of your previous undergrads who came to do his PhD with me, Christopher Walker. He's now at MUSC doing his postdoc. But so he took on this task to look at how alcohol influenced these interactions. And what we found was that we see decoupling of these PAPs, these peripheral astrocyte processes, from the synapses at the 10th dose, but even more so after this washout period. And if you just focus on the blue and green bar for a second, these are animals that didn't get alcohol. And you'll notice that they're continuing to co-localize across this period of time. So it looks like the maturation of the tripartite synapse is actually occurring across a period of time that is much longer and takes much longer than we ever imagined originally. So in terms of, I don't think it's semantics, but I think it's important to say we don't know whether it's actually decoupling or whether we're preventing coupling across this maturation period. I will refer to it as decoupling for simplicity's sake. So very little is known about what happens to these tripartite synapses and when they're fully matured. So this is something that we're starting to look into using spatial transcriptomics. But the 3D reconstruction of these astrocytes is pretty labor intensive. So we decided to switch to super resolution microscopy and essentially repeated the experiment using simple IHC really for a pre and postsynaptic marker and a PAP. And we're able to demonstrate not only that we're still seeing decoupling after this abstinence period, but now we can actually get true measurements between these synapses and the PAPs and the pre and postsynaptic terminal to understand whether the decoupling of this synapse, of the PAP is influencing the proximity of the actual synapse itself. And what we're beginning to understand is that, and this has already been published, is that really the proximity and the vulnerability of these tripartite synapses is highly dependent on the brain region and even the subregion. So if you look at the orbitofrontal cortex, ventral versus lateral, somewhat similar functions, but definitely in the same region of the brain, obviously based on their names, but we see very different effects of alcohol in the ventral versus lateral. So we can't be general about how alcohol is affecting the brain as a whole. So the question is, what is the relevance of this PAP synaptic decoupling? Well, it's really important because, especially at excitatory synapses, when glutamate is released, one of the really important roles of these astrocytes is to suck up this glutamate and remove it from the synaptic cleft to prevent hyperexcitability. So if anyone has experience with epilepsy, what you see is excessive glutamate release. And one of the really nice ways of being able to control that is with gabapentin. And one of gabapentin's targets is alpha-2 delta-1 receptor that is located on the neurons, but it actually blocks astrocyte-secreted factors from interacting with those receptors. So sucking up this glutamate, removing it from the synaptic cleft is really important, and essentially it gets recycled as glutamine back to the presynaptic terminal. But astrocytes don't act the same way as neurons. They're not electrochemically active in the same way that neurons are. So a lot of the signaling that happens through these astrocytes is via calcium propagation. So when glutamate interacts with these glutamatergic receptors on the membrane, you'll see a propagation of this calcium signaling. And one of the things that can do is regulate gliotransmitter release back onto the presynaptic terminal, which then can regulate neuronal activity. So if we lose proximity of that PAP from the synapse, you can imagine the diffusion of that glutamate is going to have to go much, much further to signal to the astrocyte what it needs to do to help regulate that neuron. So we wondered whether a loss of PAP synaptic proximity could indeed compromise astrocyte calcium responsivity to neuronal activation. This work was taken on by Christopher Walker, and when he graduated, Olivia Coulter, another graduate student, took on this project. And essentially what we're able to do is go back to these acute hippocampal slices, stimulate the Schaefer collaterals in the hippocampus, so this circuit, very defined circuit in the hippocampus, and stimulate the neurons and see how the astrocytes are responding using this adeno-associated virus that is intracranial injected and responds to calcium specifically in astrocytes. And what we see is this decrease in calcium responsivity to neuronal stimulation after alcohol. And we were super excited about this because this confirmed that that it was obviously this decoupling that was driving this effect. But there was one experiment that we still needed to do to confirm that, and that was to make sure that glutamate wasn't reaching those PAPs appropriately, and that was the connection. Well, it turns out that when we use a sensor to look at glutamate on the surface of the astrocyte, so glutamate that is extracellular, but it's interacting with these surface sensors, we actually see an increase in glutamate interacting with the surfaces despite the loss of proximity. So it appears that even though the glutamate is there, the astrocytes can't respond appropriately. So the next simple question was, well, is this simply because we have a loss of transporters and receptors on the PAP membrane? So Olivia went ahead and did this 3D reconstruction of these astrocytes again, and using IMERA software was able to distinguish between receptors and transporters that are actually physically located on the cell membrane versus the ones that are internalized. She didn't find any changes in transporter expression, membrane-bound or internalized, so they're not changing. She sent me data last night, but I didn't have time to put it in here, but she has now finished the receptor expression work, and we have no changes in receptor expression either. Now, that doesn't mean that functionally they're working appropriately, but they're at least in place to do their job. So we don't think that it's due to any changes on the surface membrane in terms of these receptors and transporters. But why do we even care, right? How is this relevant translationally? So we really wanted to bring it back to behavior, which I absolutely love, and we decided to use a contextual fear conditioning paradigm. And essentially, you take the rat, you place them in an operant chamber, you allow them about 10 minutes to collect baseline data on how much they're freezing in this new environment, and then you give them 3 foot shocks. Nothing that causes damage, but enough to make them freeze. And the more foot shocks they get, the more they freeze out of fear. You then take them out of the box, put them back into the same environment on day 2, and you look at how much information they've retained. If they've retained the information, they're going to freeze a lot at the beginning, and then they're going to realize that the foot shocks aren't coming, so their freezing is going to decline, and that's part of the extinction phase. And then by day 3, this is a typo, sorry about that, and then by day 3, it's all extinction. So the idea is that they should really not be freezing very much if they're learning and extinguishing fear appropriately. So what we did here was we tried to essentially recover astrocyte calcium signaling in this model because we know that alcohol impairs this fear acquisition in these animals. So that's day 1 of learning. So we used something called DREAD. So this is a designer receptor. We intracranially inject it like many of our studies. And it expresses specifically in astrocytes. And what it allows us to do is use a designer drug to activate these receptors that will boost astrocyte calcium signaling specifically. So essentially, we're trying to recover astrocyte function. So we inject this DREAD. We go through our typical paradigm, and then we do fear conditioning in IHC. And this is just showing you the expression that we have in red. But essentially what we see when we run these animals through fear conditioning is that if these animals have been exposed to alcohol only during adolescence, so remember this is after the abstinence period, so this is persistent, we see that this fear learning there's a fear learning deficit in these animals that have been exposed to binge alcohol. However, if you artificially activate these astrocytes to recover astrocyte calcium signaling, we can completely recover this behavior. So we're not targeting neurons at all here. This is all astrocyte calcium activity. And you'll notice that even on day 2, on this retention extinction day, that there's quite an interesting trend in the data in which the animals that received the DREAD activation actually are retaining that information a little better, and but also extinguishing a little slower. But that makes sense because they're retaining the information. So it really looks like astrocyte activity can really boost cognition. So as we think about this, so we're able to recover astrocyte calcium signaling, but what is happening downstream of that? Am I running out of time? I have no clock up here.
We got a few minutes, and then
Okay.
questions.
Okay, all right, so I will speed through. I don't think that I have that much left. Okay, so we were really interested in what's happening downstream. You know, we can't inject these chemogenetic factors into a human brain, so we really want to understand the mechanism. So we know that calcium propagation is really important for this gliotransmitter release, and we know that there's excessive glutamate that's present in the synaptic cleft from the previous study. So we wondered whether this loss of calcium signaling means that we have less gliotransmitter release to dampen down neuronal activity. And one of the ones that is really important for this is adenosine. So if we boost calcium signaling in the astrocyte, are we indeed boosting adenosine release to modulate neuronal activity? So we repeated the experiment exactly the same way as we did before, with the addition of an adenosine sensor located in the hippocampus as well, and repeated this fear conditioning task. So I should mention, with this sensor, we also have to implant a fiber in the brain as well. It doesn't look pleasant, but this is like my dream experiment. The fact that we can combine behavior with real-time changes in brain activity. So we implant these fibers, and what it allows us to do is look at adenosine release in response to both the designer drug and in response to the alcohol. So these are just some of the traces that we get during the fear conditioning paradigm. And as you'll see here, we're looking at adenosine release. And if you just focus on the blue and red bar for now, you'll see that after ethanol exposure, the animals look fine at this baseline pre-freezing stage of this session. But as you see, as they progress, you start to see this depletion of adenosine in the alcohol-exposed animals. If you use a designer drug to boost astrocyte calcium signaling, we have this really robust increase in adenosine release, suggesting that indeed this calcium propagation is critical for adenosine release to then modulate neuronal activity. And this fits really, really nicely with this freezing behavior that we see here too. So Olivia went back because we were curious, because adenosine can actually work in an excitatory or an inhibitory manner, depending on the type of receptor that is there. And it turns out that in the naive animals that haven't, I say naive, the ones that haven't gone through behavior but have received either water or ethanol, actually have an increase in A2A receptors and ENT2 transporters in this region, suggesting that, you know, as typically is seen in the brain, when there's a deficit in a signal, like a loss of adenosine signaling, that the system is going to try and calibrate and upregulate receptors to try and reach homeostasis. So I think this also really supports this idea that we have this. I think that isn't reflected in the freezing behavior because we're hitting a ceiling effect. So finally, I just want to mention one other thing that Olivia wondered about, and that was, well, we're putting these chemogenetic, these DREADS injections into the dorsal hippocampus, but, you know, this is a network, right? All of these things are connected. This fear network is really complex, much more complex than I'm putting up here, but this is our simplified version. If we're influencing the astrocytes in dorsal hippocampus, is it possible that we could be influencing the entire fear network? And it turns out that she was absolutely right. And this has never been shown before. This is not published. But what we're able to see is that astrocytes that are activated in dorsal hippocampus can influence neuronal and astrocyte activity through that entire network. That's pretty powerful, right? And after alcohol exposure, what she does see is that there are differential effects of alcohol in males and females in terms of blunting of astrocyte responsivity in prefrontal cortex for males and amygdala for females. It actually perfectly represents the types of behavioral deficits that we see in males versus females. So that was really exciting. So I'm going to stop there. This is kind of a summary of all the things that we're interested in, but really what we want to get down to is how can we understand how these drugs are working in a way that we can transition over to some kind of therapeutic that targets astrocytes. And I'm going to skip these. If you have questions about them, I'll take them. I just want to acknowledge my lab and collaborators and past members. And these are the current amazing students in my lab and my two collaborators on this project, my husband, who has no choice, and Brandon Henderson with his two beautiful twins. And I'll take questions. Thank you.
Thank you, Dr. Risher. That was an awesome talk. In terms of drug discovery and even some of the medications that are already available clinically, medications that are used for alcohol withdrawal, things like acamprosate and other medications. Is there any medication that is used clinically that is known to help with this astrocyte calcium signaling or help with the behavioral side of the experiments that you've shown?
That's a great question, and no one has looked because astrocytes aren't important. No one has really looked at those things. Some of the data, we're kind of digging into why calcium propagation is impaired, and we're getting to the point where it's looking like mitochondrial bioenergetics is a critical component of that. They really do work hand in hand, which kind of brings me to some drugs that are on the market but aren't used for alcohol use, like GLP-1 agonists. So some very recent work has demonstrated that they're pretty effective in lowering drinking in humans, and it's been replicated in rodent models as well. But it also improves bioenergetic function. So we're looking at, so we're planning to look at GLP-1 and mito-NEAT as specific targets for that bioenergetic function. Now, I don't think that, I think they're going to hit everything, not just astrocytes, but no, the ones that are currently on the market have not been looked at, and we would love to kind of look at those things for sure. Yes.
Thank you for the great talk, and it makes me very happy to see astrocytes are getting some attention. I think neurons have had the limelight for the long. So I've worked with podocytes, and I like to think them, podocytes are sort of astrocytes equivalent in the kidney. They help in the filtration barrier versus it's the blood-brain barrier in the brain. So outside of synaptic health, do you know if just the decline in blood-brain barrier because of astrocyte dysfunction leads to any of effects of alcohol that you see?
So we don't, and it is on our list of things to do, especially if we're seeing retraction of the PAPs from the synapses. We assume that we're also going to see retraction of those end feet from the vasculature as well. And that obviously that's going to have an impact on blood-brain barrier. We are working with a collaborator from WVU, Jason Huber, who's more of a blood-brain barrier guy, to kind of look at those things. And we recently optimized some assays to do the dextran experiments to see whether there's compromise just on a very general level, but it's in its early stages. But yeah, I definitely think the blood-brain barrier is going to be critically important, and I think it goes even beyond that though. So I'm actually going to go back, because this is my sneaky way of talking about things that I don't have time to talk about. But one of the things over here, there are lots of things in adolescence and early childhood, in adverse childhood events that are going to influence drinking to begin with. But what is happening at the end? We know that astrocyte calcium signaling is impaired, but that also happens in aging. We're seeing this in early adulthood. So I think no one quit yet, but I decided that what we really needed to do, because we only have snapshots, is dose these animals with binge alcohol during this teen, early 20s period, and then switch to moderate drinking through the rest of their lifespan. So they start at postnatal day 30, they end around postnatal day 500, and yes, no one has quit. So we dose these animals 5 days a week, and we're on our fourth cohort now, and what we realized was, we were taking blood and brain, but everything else in these valuable animals is just going to waste. So I went around the whole department and I was like, do you want some bones? Do you want some liver? And by doing so, we now have this group that are looking at all of these different things in exactly the same animal, which is, very rare. But so we're trying to look at systemic inflammation and how blood-brain barrier may be influenced from both directions, from inflammation in the brain or changes in the brain, but also things externally. We're looking at microbiome and its relationship to the gut. We're also looking at telomeres. I will tell you that data does not look good for those of you that are continuing to drink through the rest of your lifespan. But you're probably going to be okay if you started later in life. But that data has not been published yet. But so it's really exciting, because we start to bring the entire body together and not just think about this as a brain issue, right? So yeah, I don't know whether I answered your question. Okay.
Thank you very much. Just as a follow-up question to what you have up on the screen now, considering binge drinking, particularly amongst adolescents, with, let's say, early adulthood, like what you have in the university student population, what would you think, or what would you predict as the long-term sequelae of this binge drinking in the hippocampus, knowing that that's the brain area that is particularly implicated in or involved in learning a new memory?
So we know that during acute exposure, regardless of age, that you're going to have deficits in learning and memory processes acutely, and it's much worse the younger you are. A hangover isn't going to help, right? But, just at a baseline level. So if we look further out, the working memory component that the hippocampus is really driving is a little complicated. So we do, and the data is mixed. So in the human data, there does seem to be some recovery over extended abstinence of that of working memory. But it really depends on how much damage is done. And the damage that is done is really dependent on the blood alcohol levels that you're hitting. So if you're hitting repeatedly something that is in the 190s and higher, if you're hitting 200 mg per deciliter on a regular basis, you're going to end up with neuronal death. So the blood alcohol concentration is a really, really critical factor for that. So if you're hitting those really high levels, the likelihood of your brain recovering fully in terms of working memory is going to be lower. If you're a more moderate drinker, you're not going to get the neuronal death or the inflammation, and the recovery is going to be much better. I hope that answers your question. Okay.
Thank you, Dr. Risher. Thank you, Dr. Eric Worth. We move forward with the UPIKE faculty oral presentations. I hope everybody is here, all the faculty members who are supposed to present. We have a great mix of talks representing the many different schools on campus. Talks from the optometry school, osteopathic medical school, business school, undergrad school, and the dental school. So I hope that it's going to be interesting for everyone that is here. So the first talk or first speakers are from the Kentucky College of Optometry, Doctors Amanda Blankenship and Emily Koenig. All right. And quick reminder, 15 minutes for your talks, please. So there is a timer here as well, okay? I might just knock on the table when 1 minute is done. All right?
I guess it's still morning. Good morning. If you're like me, we've already had a whole day before we got here, but thank you so much for having us. It is an honor to be here among all of you. I am humbled by what our presenter has done and what they're continuing to do in their work and what everyone is displayed around us. Just thank you. We are humbled and very appreciative to be among all of you. This presentation, I'm just doing the introduction. It's about building bridges. I want to just take a moment for everyone to just close their eyes. Close their eyes, and I want you to think about someone who changed the course of your trajectory, who influenced you, who guided you, who gave you advice. Think of that person in your mind's eye, how treasured that relationship was. Someone who shared their knowledge with you and imparted that knowledge. It could be a teacher, a family member, someone who made a difference in your life by engaging with you. Do you have that picture in your mind of that person? That was an example of a really incredible, excellent mentor in your life. How important that person was. Now you can open your eyes. Think about that relationship and how that has influenced you. And we wanted to do a little bit of research on mentorship because, as you know, you're here today in part because of that person. The things that happened to you positively and made a difference in your heart, it was that person. And because of that important mentorship relationship that we've also experienced in our lives, we wanted to think about an innovative approach and research that innovative approach to mentorship. We have nothing to disclose. And now I'll turn that over to my coworker and friend, Dr. Kennan.
Good morning. So like Dr. Blankenship said, our presentation today is really about mentorship and our survey study that we did with this. So here at the University of Pikeville, we have a wealth and a myriad of teaching experience and pedagogical knowledge, spanning from the expert who may have been in a higher education role for many, many years, to the novice who may be just starting out in their first higher education role. Nevertheless, we believe that every faculty member, every educator has something that they bring to the table of education. Previously published data has revealed that having effective mentorship is really critical for helping fill gaps in faculty pedagogy and also to help move and advance their careers forward. So as you may know, K through 12 educators, when they are trained, when they go to school, they receive formal training to complete education to develop their pedagogy and develop their skills. Whereas us in higher education, are responsible for the development of those skills. We know we complete a PhD or a doctoral graduate program to kind of get us here to these fields, but we don't necessarily receive formal training and instruction. So there may be gaps in our teaching methodologies and instructional strategies. So having new faculty mentorship programs such as we do here at UPIKE is really helpful to help us develop our pedagogy. But as you know, most mentoring programs typically end after the first year of being on faculty. And I'll be the first to say, as I'm wrapping up my second year here teaching, there's a lot to learn after the first year. So we really wanted to look at, an additional mentorship program that we could bring to extend beyond this first year, because we can continue to learn and grow and develop over the course of our professional careers. So what we wanted to really look at in the study, we wanted to take it a step further. A lot of times our mentorship and our development occurs within our own departments. My mentor, Dr. Blankenship, we both work at KICO. But what we really wanted to look at here was kind of breaking down these boxes and silos that we kind of get put in, or we put ourselves in, and we speak to the same people every day, eat lunch with the same people every day. We really wanted to create these partnerships and mentorships across disciplines to see what we could learn from those disciplines as well. Because maybe we don't teach the exact same material as somebody in social work or potentially nursing, but we can learn from their instructional strategies and how they teach their students and take that to how we teach our students as well. Sorry, having some technical difficulties. Okay, so here were our goals for the study. We wanted to create an environment of collaboration and professional growth, strengthen UPIKE's sense of community by working across campus, empower our faculty to model interprofessional education for our professional students. We ask our students to participate in these types of activities throughout the year, and this is just another way that we can model this for our students, and also allow our faculty to acquire improved pedagogy through peer evaluation. So for our study design, we conducted the study last spring over the course of a semester, and we paired faculty volunteers from the Kentucky College of Osteopathic Medicine, the Kentucky College of Optometry, Elliott School of Nursing, and the School of Social Work. And we asked these pairs to informally observe their partners' recorded lectures, view their Canvas courses, and meet together periodically to discuss topics such as their educational approaches, assessment tools, and course building techniques. So we did ask them to meet at least twice throughout the semester, and each participant received a $100 stipend to facilitate these meetings as well. And then in order to kind of look at the study at the end of the semester, we asked the participants to complete a survey detailing their experience in the program. Sorry, I can get it to advance to the next slide. I'm sorry.
You can use this thing as well.
Yes, okay, cool.
So, yeah.
Thank you.
All right, so how about just use, yes.
Okay, perfect.
Let's use that.
Okay.
Very good. Thank you.
All right, so we had 12 volunteer participants in the study, and at the end of the semester, we conducted the survey, and we did have an 100% response rate from our participants. As far as the way we conducted the survey, it contained 14 questions. The first 11 questions were Likert scale questions, so we presented the participant with a statement, and they had to say whether they strongly disagreed, were neutral, agreed, or strongly agreed with the statement. So 1 being strongly disagree, 5 being strongly agree. At the end of the survey, we also had 3 open-ended questions, which allow the participants to describe the strengths of the project, the weaknesses of the project, and opportunities for improvement as well. So I do have some summary slides kind of summarizing our findings from the study, but these are some of our survey questions that we included. And you can see here on the slide, I've included the mean response for each question, how many people strongly agreed, disagreed, and strongly disagreed. For the purposes of this presentation, we didn't include the neutral responses. So here's some of the survey questions. Again, I'll summarize here shortly. And here are the other ones that we included as well. And overall, if you look at the survey questions, you may notice that I also included the p-values, and they're not necessarily particularly strong for this study, but do keep in mind we had a pretty low sample size. So overall, this project had weak statistical power given our low sample size. But overall, the participants seemed to have a positive experience, as most of the participants agreed or strongly agreed that they had a comfortable teaching and learning experience. The project encouraged sharing and helpfulness. They felt that the information shared by their partner was useful, and they planned to apply what they gained from the partnership to their own teaching methodologies. The project seemed to set an example of positive interprofessional collaboration for their students, allowed for professional development through collaboration with another UPIKE faculty member, and most participants agreed or strongly agreed that they would participate in this type of project again. So considering the statistics and the survey comments regarding the strengths, weaknesses, and opportunities to improve the project, we conclude that overall, even though this survey did have weak statistical power, we still did receive positive survey results that'll offer us valuable insights for potentially, completing another type of program such as this in the future. So most participants found the project and their partnerships helpful, believed that the information shared between the partners was useful, planned to apply what they learned to their teaching practices, and benefited from the professional development that was provided. So as mentioned earlier, our last question on the survey gave an opportunity for the participants to provide areas for improvement in this type of project going forward. And here were kind of the most common things that we saw. So one of those being scheduling regular feedback sessions between partners, as not every single partner pair was able to meet more than one time throughout the semester. Next, intentionally matching partners for peer evaluations, as given the difference in schedules, it was a little difficult for some people to receive a peer evaluation, which we know is important for things such as our portfolios. Providing a checklist of activities. Some participants were kind of hoping or would appreciated kind of like a more formal list or checklist of things for them to discuss and talk about and do. But some participants did appreciate the flexibility to discuss their own topics of interest and things that they felt like they needed the most help with. Another suggestion was to organize a final group Zoom meeting for experience sharing at the very end of the project, so everyone could discuss what they learned and what they gained from their partnership and how they plan to apply that in the future with their teaching methodologies. And then lastly, extending the project duration to a year. As we all know, some semesters are more hectic than others, so some people found it a little bit difficult to meet with their partner as much as they would have liked to. So by extending the project duration to the year, may have given the opportunity for more meetings, and also maybe given the opportunity to look at different types of courses and how that instructor approached teaching those different types of classes. So overall, we feel that this type of professional development model can be a promising supplement to other college professional development programs, Certainly, we don't feel that this isn't a replacement for the new faculty mentorship program, but could be a promising supplement or addition. So we can continue developing these partnerships across campus over time and continue to grow and develop as faculty. And we feel that this research provides a useful foundation for further studies in this field and also potentially future projects in the future such as this here at UPIKE as well. So we would like to thank all of our faculty volunteers that we had from the Kentucky College of Osteopathic Medicine, Kentucky College of Optometry, Elliott School of Nursing, and the School of Social Work who participated in our study. And we'd also like to thank Dr. Meg Seidel and her office for their assistance with the statistics. All right. Do you have any questions for Dr. Blankenship or myself? I'll take those.
They're good. All right. Thank everyone. And again, I see some of the people who volunteered faculty for this research. Thank you, guys. We could have done it without you. Thank y'all.
Thank you so much, Doctors Blankenship and Koenig. So next up, we have Dr. Kevin Wang from the College of Arts and Sciences. Hey, Dr. Wang. So you can move the slides from here and 15 minutes, and just see that the mic is okay if you want to change it.
Okay, thank you. Thank you all. Yeah, my topic is we use plant to produce, actually here is blood clot dissolving proteins, blood clot dissolving proteins. Actually, I would like to talk about, we use plant as a gram bioreactor at UPIKE for our undergraduate, both undergraduate, graduate, and medical school. Because usually when we do the biopharmaceutical imaging editing or synthetic biology, yes, we use animal cell or microbial bioreactor, but for animal cells, it's very expensive. Also, animal cell line itself, it is very difficult to maintain. For microbial bioreactor, yes, it's less expensive, but remember that it is very difficult to see the, we call the morphological difference. Yeah, especially for our undergraduate students. If for our undergraduate students, they don't feel or they cannot see, yes, very difficult for undergraduate students to involve the research. The picture is Alexa Dickey. Yeah, she was she was working with me long time ago, and now she published several as a first author and co-author, and now she is a real doctor of family medicine. So that's why we want to try to build a plant biotechnology at UPIKE, because plant is safe, is low cost, and also easily reproducible, and you can touch and feel. And in here, we have already developed the transient expression, means rapid. Transient means rapid, yeah. Like usually it's 3 to 7 days we can get results. Here you can see the, this one. Where's the, it doesn't matter. You can see the gram GFP, yeah, glowing. Another one is stable transmission. Yeah, maybe next year we will do that. We will use like tissue culture, yeah, sterile technologies for a couple months and get the plant. Because non-plant biotechnology can do the cutting edge biotech, what I said is for biopharmaceutical, you cannot imagine plant can produce biopharmaceutical. Actually, it is a chance. And synthetic biology and gene editing are cutting edge. That's why, yeah, our project is awards has been recognized by our NIH, NSF, or KY Embry, collaborated with our medical school professors, Dr. Kinistin and Dr. Avlabi. So we secured the grant from KY Embry for course-based undergraduate research experience awards for three years. And also here is my student. Maybe some students know that Kali Hall, we demonstrate the algorithm like for transient expression technologies. Yeah, we use 3 to 7 days to produce reagents. Yes, commercial enzyme. What I said is a platform for our UPIKE for functional genomics, for biological research tools, and also for educational purpose. Yeah, educational is our priority for our universities, especially for our undergraduate students. And we submitted the manuscript already is under peer review process. And here you can see, yeah, for using transient expression, yeah, we can see design. You can design any proteins. You can design synthetic biology. You can design the cutting edge gene editing. Yeah, you know gene editing, but you do not know how to do that. So we can use plant to do that. It's safe and rapid. Yes, and also, yeah, like what I said, rapid means after 3 days you get results. It will last about a month. Yeah, a month. All the tubes, you can see that. You can see, yeah. Just like drugs, functional genomics, yeah, functional genomics and gene editing analyze. We call say design, build, and test and learn. Once we learn something, we do it again. And also, this picture is submitted to a manuscript. Is in the peer review process. Here is the built foundation of rapid protein production platform for UPIKE. I would like to thank all my students. Yeah, they spend lots of time on that. Yes, and get product and get results, get data for publications. That's why for undergraduate students, it is very important to let them to see and feel the cutting edge technologies. Sure, we appreciate the traditional lab, just traditional, but for cutting edge, we need to get access. So also in the future, we will do the, not in the future, we will do the, for example, letters, not just like the tobacco. Actually, we use tobacco as a model plants to do a biopharmaceutical gene editing and synthetic biology, but also we can use lettuce, we can use spinach, and also other plants. That's why it's good for our students to test. It's safe. So in the future, as I mentioned, to do the using plant to do the we call cutting-edge biotechnology. It is a tool. It is a tool, especially for our undergraduate students. Yes. Yeah, this is planned as biopharmaceutical factory. It is a trend in the future. Actually, it is a trend now. Lots of giant biopharmaceuticals that are using the plant to produce biopharmaceuticals, because all biopharmaceuticals, maybe you don't realize, very expensive, sometimes $1 million program, So we can do this. So I would like to talk about just one slide. Yeah, we use plant to produce blood clot dissolving proteins in plants. So why we want to use this? Because if you have stroke, if you have cardiovascular disease, if you have a heart attack, remember the drugs are very expensive. We call recombinant proteins. Over $10,000 per gram. If you buy one, for example, number one Alteplase from our PMC Parkville Medical Center, is about $30,000 per gram. Yes. So very expensive, yeah. Because why? Because it can dissolve. Now, all those recombinant biologics are from CHO cell, animal cells. So it can dissolve, kill cells. That's why we want to use plant. So actually, you can see here, plant-derived proteins can dissolve blood clot. Yes, and also because this is very powerful. For example, when we introduce all those genes into plants, the proteins can dissolve leaves. Leaves means it is very good. It is very good. We have good results. Yes, and also this plant-derived blood clot dissolving proteins received an award, his most innovative science poster presentations in Washington, D.C. last year. So it's top one out of 517 posters. So here is our UPIKE, yes. And also in the future, because under Dr. Eric Worth's lead, we have REI, Research Center. Research and Entrepreneurship Incubator, yes. So because it is very good for us, because you know why, for example, if we have transient expression of stable biotechnologies next year, we will build. And couple of days we can get the results, plant factory. Yeah, now we are building the our greenhouse in REI, and 2 to 3 days and get characterizations for both undergraduate and graduate research. And all for our medical professors, if you need functional proteins. And also why we like plant, as mentioned, plant cannot, even plant, we use plant virus, okay. Plant virus cannot affect human beings. For example, you eat lots of fresh food, fresh fruits, but you cannot get sick. Another one is, once we have greenhouse, we have a safety standard. Yes, we can control that. We call controlled environment agricultures for gene editing and synthetic biology. So now, like right now, I have to go to my office lab every day, especially night. You can see the GFP. So this year we 100% from UPIKE, because we use these systems. We submitted a gene sequence character 1 for stroke, kidney, and cardiovascular disease in deposit in gene bank. Is Carly Taggett, Carly Hall, and Holly Jordan, Gabby Hall, and Peyton. Yeah, they're all here, all 100% from our UPIKE. And also last year we deposit also 100% from our UPIKE. It's nanokinase genes for the cardiovascular disease. And also we have already submitted several manuscripts and in a peer review process. I just got a email from ACS Omega editor, and he said he said we are thrilled to receive it and are genuinely excited about UPIKE work. We believe it holds significant potential. It's just a long email. So they never expected our UPIKE undergraduate student can do such a good job. So I would like to thank KY Embry support, and they recognize our work projects. And also I would like to thank all my students, and especially thank Dr. Eric Worth. Without Dr. Eric Worth's help, it's very difficult to move forward. And we are blessed. We have 2 Dr. Worths helping our UPIKE. And also, I would like to thank all my students. They work very hard. I'm not sure. Yeah, sometimes even I told them just to work on it. Yeah, start. Yeah, start. You don't need understand. First week, you don't need to understand. You have to work on it and then understand slowly. Yeah, gradually. Yes. And here are my students today. Thank you all. If you have any question, let me know.
Yes. Okay, thank you so much. Excellent talk. Yeah, I really appreciate it. I mean, it's exciting, and I appreciate the work you've done with discovery of the synthetic nattokinase. Is that it?
Yes.
That's been deposited in GeneBank. I was really excited, when I saw that work has been done and UPIKE is represented in GeneBank. So my question is, do you plan to go further with drug development, and how do you intend to validate your studies? Will this synthetic nattokinase really be, is it possible that it will be used in human? Do you plan to use animal models to validate your work?
For this plant-derived nattokinase, this is just one of our projects. We compare to the commercial nattokinase. The activity is very similar. We have already statistically similar. For the commercialized, I'm not sure. For further development, you can ask Dr. Eric Ross, because he's the director of innovation and development. Yes, for this one, I just follow Dr. What's your direction? Because we have lots of products, yeah, potential products.
Thank you.
So that's nattokinase, that's the one. We are going to produce like TPA. I guess some people know TPA, red place. Yes, you know TPA. Yes, for stroke. Yeah, and yep, this one we have already produced already. Yes, this one. So we want to compare, because now why I don't have data, because the commercial products are very expensive. I'm still looking for the less expensive, maybe donation, maybe reagents from my collaborators. Yes, once we receive the commercial one, we call comparisons, we will publish.
Thank you, Dr. Wong, for the talk.
Yeah.
I have a question. So, right now, this tenecteplase and alteplase, they're produced in mammalian cell lines, right?
Yes.
So do you think, would that affect the immunogenicity? Because the allergic reactions to these drugs are also a problem. So do you think, or have they seen in any other plant-produced medications, how does the immunogenicity or allergic reactions compare when you produce them in plants versus animals?
Yes, it is immune response. Why we can say this is a new trend? Because usually the plant produce, the yield is very, very low. Purification it does not mean like we have immune system against the plant, because we eat plant every day. Yes, we eat plant every day. You don't have immune response. Actually, oral drug is always safer than injection. Yes, this one. Yes, so this one. So why now it's a trend? Because usually the yield is very low. We cannot purify. Now the yield is very high, 1 gram per kilogram. Yes, so that's why it's a new trend. We compared the safety issue, the safety efficacy, the safety and efficacy is similar, or we call biosimilar. And also sometimes we will screen, we call biobetter. Even the activity is much better than, because oral delivery, sometimes injection oral delivery is much better than your injections. Because the plant cell, maybe some students, they don't know that plant cell is a high advanced cell, eukaryotic cells. They have more complex mechanisms to modify, to do the post-translational modifications. That's why now it's a new trend for even giant biopharmaceutical companies. So in the future, in the very near future, we will do the oral delivery. We will do, for example, for the this one, you don't need injection. Yes, you need the oral deliveries, because injection means if I have stroke right now, we only have 1 hour. We call 1 hour window to send me to the medical. Sure, here is very close, but how about you live far away? You know that, yes? You know that, yes? So that's why we want to make an oral one. So shelf life is about 3 years. Actually, shelf life means what? Room temperature. Thank you so much. If you have more questions, let me know. And also, I have our students. You can see that posters.
Thank you so much, Dr. Wang. Thank you so much. Okay, next up, this computer. Okay, finally it's here. All right. So next we have Dr. Praveen Goraja from the Coleman College of Business. Dr. Goraja. So make sure you adjust the mic. Yeah. Time 15 minutes, and you can move the slides from here.
Sure.
Right, thank you.
Thank you. Good morning, everyone. So today, my topic is about engaging the next generation in rural Appalachia with all the technologies that each of you here are touched by, right? You know, people talk about, irrespective of what field you're from, you all, as a part of your discussion every day, might be talking something about related to AI and how it's touching each of your fields, right? So today my topic would touch the same, but I would try to bring in different perspectives. Most of it is my perspective of different things, and I would love to hear from you as well of some of the things. So let's make it interactive so that we can learn and exchange ideas, right? So to develop the next generation workforce, there are few aspects of technology that are highly influencing these things. One is artificial intelligence. Of course, your data is read and analyzed in ways that you have never imagined. Under the umbrella of data science, we all have these, right? And then one of them is data analytics. So they go deep and deep into your data to study. Are they good enough to study the patterns? So we will try to find. And then micro-credentials. At least in the undergrad education, micro-credentials has become a big thing of today. So I'm sure that medical, like on the KICOM side, you have board exams. So, and then that's when you get your board, like license and stuff. So these are like small type licenses to show that you have the skills to prove, for your future job that you would have, right? So you all agree that the student landscape is evolving so much. And if you're an educator like me, you would definitely understand that, that aspect of it. So one of them is digital search. 95% of US teens now have smartphone access, which was before, probably 73%. Knowingly, unknowingly, in classrooms, they keep touching some kind of digital, asset, right? It could be your laptop, it could be your iPad, it could be your phone. So their attention span, in previous studies, they used to say if you can't, catch an attention of a student, then, they would be distracted by something else. That is more true than ever before because that attention span has come down even further, right? And the technology that is helping you is also ruining some of the stuff that, like we as educators would like to see in students. The most important of all is originality, right? Your thoughts are influenced by something, right? You always try to search for things rather than trying to search, or think through and bring the original thought from yourself. I think technology should be used as a complementing thing, but not as your first, reach, right? So I'm going to go over that within the limited time that I have. Interactive learning. Today, if you don't, let the students, be a part of your interactive learning, again, as I told you, they don't pay attention. So that is what we are dealing with. That has been a struggle. I think that will continue to be a struggle. And then I think, we are going more towards interactive learning. I'm going to talk about it as well. Health fields evolution, right? You all realize that I'm not sure what type of technologies is being used here, but at least so far I've heard from some of the faculty that they would love to have some kind of virtual reality, right? Augmented reality and mixed reality used in your classrooms. I'm sure that would be the future. If not now, it will be implemented, pretty soon. Do you guys use any of the VR, like MR technologies in your classroom? Like virtual reality, augmented reality in the classroom? That's good to know. Dental school, the new upcoming, dental school would be using it in their school. So that would be a thing in health fields, right? You must have heard about it. I'm sure if not implemented, faculty must have talked about it in the classrooms. They must have shown you about it. So there's a big evolution coming, so that will also change the student landscape. Global tech influence, right? Like the AI, VR, quantum chips. Anybody saw recently these tech giants has said that, hey, we have quantum chips now. What does it do to everything that is out there? Anybody, quantum chips, have you read the news? What does quantum chips do? We didn't have the computing power before to do certain, tasks, but now you have the power to do it, and that is going to disrupt everything that you have thought about. For example, if you ever thought your password is secure because it's encrypted with 256-bit encryption, but now with quantum chips, they can break and tell your passwords. So there is good and bad part of this. And then before even you can think and, attempt to build frameworks around this, it's already there. Today, the main problem is you have technologies without proper frameworks, proper guidelines, right? By the time you frame it, it's already has changed. So connectivity in rural Appalachia has changed so much. When I came here 4 years ago, the biggest problem I used to talk about is, hey, our internet speeds are not fast enough, right? But then after that, I saw there were so many federal and state projects that were going on, and broadband expansion has furthered some of the connectivity in this region of Appalachia. I'm sure in some parts of Appalachia, it's still a problem, but significantly it has improved. And I've seen it because I was part of one of the groups where they were talking about we got some $1 million of grant to improve broadband within this region. So that would open up so much of the things that we were not able to do before. So there is infrastructure gains that I was talking about, new learning horizon, right? So things that we were not able to do because of the latency, because we were not able to use those tools even though we want to, because your internet speeds are not good enough. Those will change now. You would be able to use those modern technologies in this region. So then the local advantage. So connecting University of Pikeville can use this innovative, blended and fully online educational programs, which probably would have, I'm sure that online delivery is being done. It can take to the next level if UPIKE taps this market. So tech innovation and engaging digital naives, generative AI. We all talk about the word chat GPT. Can somebody tell me what is GPT? What is a GPT? Models, yes, right? So chat GPT models is nothing but generative pre-trained transformer, right? GPT means, so by the word, in my classroom, I keep telling, whenever I use explaining technical terms, I tell, in IT world, 90 to 95% of the time, by the words itself, you can predict what it means, right? So most of the time, you'll get it right. So only 5% of the time, it may not be the same. But generative, it's generating content based on pre-trained data, right? So that is what you're all using. My problem is, ChatGPT, you're all very appreciative. I'm sure most of you would be very appreciative of what, generative AI models are doing for you, right? But what if I tell you there is no original thought in it? It's always influenced by the pre-trained data, but it can build new data, but it's still influenced by what it has been trained on. The original thought always comes from humans, right? So if you are using it as your main idea, then you are using somebody else's ideas that was pre-existing. Have you ever thought about it? If yours is the original thought, then you can leverage that tool to build on it and then make it a better idea, right? But if you're using it as your main source of, data or your thought process, then I think it's flawed. I think our future generations is going to be using that and thinking that is the main source that I would go, right? They would never use, the main power that we have, right? Our imagination. Can AI dream? It can't, but you can. A lot of original thoughts came from dreams, right? So I'm trying to tell you that future generations, it's very hard to teach them and not to have them use these technologies, but I'm trying to tell you that how that student landscape is changing. I'll present you some numbers right after this slide. So why focus on AI and data analytics is because adaptive learning, AI-driven education tools, several of them out there, right? For student engagement, improving retention, and we can personalize the content. Real-time insights. When I was talking about data analytics, right? Why? They are building dashboards to study, like if at all somebody has to study what my students are doing. We used to use a tool called Neuro before to tell that who's in red, who's in green, so it can study all your data and tell these are potential students who are at high risk of dropping, right? So those kind of data is being leveraged more and more in education space. Emerging job market, 85 million jobs may be displaced by AI. It was told by this year, right? This data was from the past. I'm sure it's going beyond that probably. And 97 million new roles will be emerging. Have you ever heard about a job role called prompt engineering? Probably before. 10 years ago, was that a topic of, did people talk about it? Especially, faculty here. But now prompt engineering is a thing. What is prompt engineering all about? Anybody? What is prompt engineering? Sounds fancy, isn't it? What is it? Basically, it is nothing but, your output is as good as your input, right? If you're prompting well, you would get your output in generative AI as good, right? Prompt engineering is fine-tuning of that input so that you can benefit out of the best outputs that you can get from these models. A lot of us will do as if we are chatting with, like general chat, right? But if you try to, prompt it in a different way, your response will be different. You know that the quality of your output is always relational to your, like it's always related to how well you prompt it. That's a new job role right now. That's a new, job out there in market. So I think I have a couple more minutes. These are the numbers that I want to show after, I think the next slide is data-driven facts. So the impact of micro-credentials. What are micro-credentials? In undergrad education, now it has become, it's not about just teaching, but students are coming with a lot of anxiousness, with low confidence levels, most of it, if not everyone, but most of them. It became the educator's job to give them the confidence, give them the morale that they are really worth for everything that they're learning. But micro credentials are complementing it. It can boost their confidence by significant levels, and it's also improving their job placements as well. I'm going to present some numbers, and I'll tell you. Micro credentials are nothing but like certifications, some of the skills that you need to perform your job, right? These are some of the facts that you should pay attention to. So, rural Appalachia, 200% surge in AI and machine learning enrollments in rural learners since 2019. I am sure that this number, by the time the study was done, it's only 200%. These area micro-credentials has increased significantly. And growth of online learning, again, this is from the study in 2022. After COVID, you all know that what happened to online learning, right? Significant bump in online learning. So we in rural Appalachia, here in Eastern Kentucky is no longer limited by our physical space that we're in. We can expand our offerings, especially in our undergrad or grad education. And then students are willing to take online classes now. And then 50% growth in credential growth. Like 2023 credential engine analysis found over 50% growth in new micro-credential offerings. I'm sure if somebody is behind certification, you all saw a lot of certification is being offered now, right? I didn't see when I was using LinkedIn before, there's nothing called LinkedIn certifications. You have certifications now. You have platforms like Coursera. Just to show that, and yeah, I have one more minute. So what I'm going to do is, student views on micro-credentials, 90% of the students say micro-credentials help them stand out to employers. And 88% of employers agree these credentials strengthen their job application, right? Your job application even used to be, like seen by people before, but now it's actually being monitored by AI tools that it filters the skills matching with job description with what you posted, right? So one of the ones, AR and VR simulations in medical training, 275%. It can be up to 4 times faster than standard classroom training, with learners being 275% more confident applying skills afterwards. You all should tell me that. Is it a fact? Because one of the studies is saying that. Do you all agree with it? So the another one is 866% high demand for AI skills. You see that 866% is what? But UPIKE, I'll end with this. UPIKE can lead by example in rural Appalachia by integrating these technologies very cautiously. Like I told you, you have to be very cautious with technologies. The new word, I'll end with this. You need to learn, there is a new word coming in tech field called digital empathy. Empathy has always been related to humans. What is digital empathy? If you can take these machines to feel your feelings to a level where it can replicate like human, that's what they're doing it right now. Called a new word. You will come across this in the next few years called digital empathy. They're training these models to be digitally empathized. And actions, I would say that all those things that I talked about, without having to go with slides, we can build a future-ready Appalachia together. I think we are not limited by the place where we are at. I think these technologies are, has changed the scale, the education space completely. Thank you for your time.
Thank you so much, Dr. Garaja. Next up, we have Dr. Saima Ijaz from the Kentucky College of Osteopathic Medicine. This cursor thing is an issue. Okay. Yes, give me 1 second. Okay, finally. Okay, so I'll put this down. My mouse, we don't have a mouse, but this thing is not working. Yeah, the pointer. Okay, very good. Thank you so much. So the timer and the mic, and I'll let you know when there's 2 minutes left. Do you want to change the view, or this is okay?
I just want to have a pointer.
Pointer there, yeah, he's going to give you the pointer in 1 second.
This would work for me.
Sure.
Yeah.
Yeah, okay. All right.
So,
Thank you.
I have to go back. Okay, good morning, everyone. So today I would be basically talking about my previous postdoc research that I did before joining here, and it's about the link between Alzheimer's. I think you all are aware of one of the most common neurodegenerative disease and protein quality control. So we all know that Alzheimer's is a progressive neurodegenerative disease, and it causes gradual decline in memory and cognitive function. So it is basically characterized by the intraneuronal accumulation of neurofibrillary tangles and the extracellular accumulation of amyloid beta. So basically, these neurofibrillary tangles are formed by the hyperphosphorylated tau, which is not normal. So despite a lot of extensive research in the field of Alzheimer's disease, still we don't have a proper treatment. So some of the researchers have shown that there is disturbed protein quality control in Alzheimer's. So here you can see a study done in 2008, and they have shown that there is increased ubiquitin accumulation in the Alzheimer's model. So I would like to tell you that ubiquitin basically accumulation is one of the hallmark for the disturbed or dysfunction in the protein quality control. Okay, then another study done in 2001 shows that there is impaired dysfunction of proteasome and protein quality control in the Alzheimer's. So what protein quality control is? Basically, it is essential for the degradation of misfolding protein. Okay, so the key components for the protein quality control are UPS, that is ubiquitin proteasome system, mainly comprising of proteasome,
CMS, that is chaperone-mediated autophagy, and the macroautophagy. Okay, so if we talk about the most important part, that is the ubiquitin proteasome system, it is basically the major pathway for the protein quality control. And what happens in this system, basically a protein substrate that is misfolded needs to be degraded. So this is usually tagged by the chain of ubiquitin, and then this tagged ubiquitinated substrate is later recognized by the 19S regulatory unit of proteasome, and then later it is transferred to 20S catalytic core of proteasome for the degradation into small peptide. So now how we can modulate the proteasome activity to have a protective effect? So basically, this 20S proteasome is consisting of like 19S and 20S part, and this 19S contain lead and base. The lead contains 9 subunits of RPN, and that includes RPN6 as well that we will be talking about. Okay, so previous research has shown that the phosphorregulation of RPN6 at serine 14 basically enhances the activity of proteasome and results in the more and more degradation of misfolded protein. So basically my research is about like if we increase the phosphorylation of RPN6, that can have a protective effect, and even we can use it for the treatment of Alzheimer's itself. Okay, so we hypothesized that serine 14 RPN6 phosphorylation improves proteasome function and protects against the pathogenesis in both the brain and heart as well, especially in the tauopathy-based Alzheimer's disease mouse model that I used. Okay, so to conduct this study, we used a recently created mouse model that is known as S14A, where we replaced S14 of that RPN6 with alanine. And that S14A mutation prevented PKA-mediated phosphorylation of proteasome. Further, we use another mouse model that is the known mouse model for tauopathy-based Alzheimer's disease, that is known as PS19, where there is overexpression of human mutant tau. Okay, so what we did, we basically crossbred that S14A mouse with a PS19 mouse to get a mutant mouse where there was characteristics of Alzheimer's, but we also suppressed the proteasome function, or the protein quality control was compromised. And then we observed the effect, like what is the effect of that compromised protein quality control in our Alzheimer's mouse model. So for that, we compared wild-type PS19 and PS19 S14A that I would use from now onward a word mutant mice for the sake of time. Okay, so we did like cognitive function tests, we did motor function tests at 6 months, and then we did echocardiography at 3, 6, and 9 months of age for the heart function, and we did Western blot, confocal imaging, and the RT-qPCR to see different chemical and biological analysis. Okay, so first of all, we did the Western blot to confirm that is there really in this mouse model phosphorylation of RPN6 has been blocked or not. So you can see we did Western blot for phosphorylated RPN6, and there was complete absence of this protein that shows that the phosphorylated RPN6 is blocked, right? Okay, further, we did Western blot for the total ubiquitin and the K48 ubiquitinated conjugates. And you know, like the accumulation of these conjugates shows that there is proteasomal insufficiency. So what we observed that not only in brain, we observe significantly high protein expression of total ubiquitin and K48 ubiquitin in hippocampus, but also in heart as well. So our study basically not only linked Alzheimer's with the protein quality control, but it also linked that Alzheimer's is not only limited to the brain, but it is also linked with the heart. So that accumulation of tau and amyloid beta from the brain can circulate into the blood, and then it can accumulate in the heart and even in the other organs as well. So we wanted to observe further what is the effect of that S14A on PS19, like brain tau accumulation and also in the heart. So for that, we use different tau-based antibodies, and we observe significantly increased tau accumulation with all forms of tau in our mutant mice as compared to PS19 and the wild type. That shows that whenever there is proteasome insufficiency, there would be more and more tau accumulation. So if we go vice versa, it means if we enhance the proteasomal function, it means it might be a good target or can be used as a therapeutic option to reduce the pathogenesis of Alzheimer's. So further, like as I showed you in the previous slide, the increased accumulation in the hippocampus, in this slide you can see there was increased accumulation of tau in our mutant mice in heart as well, as compared to PS19 and wild type. Further, when we observed that there is tau accumulation in brain and as well as in heart, we wanted to see, is it affecting the heart function? So for that, we did echocardiography at 3 months, 6 months, and 9 months. And what we observed, that starting from 3 months, there was reduction in systolic function. And that is indicated by the fractional shortening, stroke volume, and ejection fraction, and as well as cardiac index. We also observed increase in left ventricular posterior wall thickness. That is one of the indicator of the hypertrophy that we observed in our mutant mice as compared to the wild type and PS19. Okay, so further, once we knew that there is disturbed cardiac function because of increased tau accumulation, we wanted to see if there is any effect on cardiac remodeling. So for that, we did histological analysis. We did picrocereus red staining while using all 3 genotypes, and we observed significantly increased picrocereus red staining in our mutant mice as compared to PS19 and wild type. Further, we wanted to observe the effect of S14A on our fetal gene activation program. So for that, we did our RT-qPCR analysis, and we observed the genetic expression of NPP-A and NPP-B, which usually encodes for, like NPP encodes for ANF, and NPP-B encodes for BNP. So basically, increased expression of these NPP A and NPP-B indicates that there is cardiac stress or cardiac injury. So that further confirmed that the disruption in proteasomal function is one of the most, it expedites the process for dysfunction of heart as well. Okay, so further, we wanted to observe if there is any effect of that S14A, like suppression of proteasome activity on PS19 mice, behavioral, like cognitive or motor functions. So for that, we did open field tests to observe their general motor activity. And we saw that the general motor activity shown by distance and velocity were comparable. There wasn't any effect of suppression of proteasome on the normal motor activity. And then we observed the time spent by the mice on the rotor rod that basically shows the function of motor activity. So we observed that our mutant mice, basically shown by red color in all the graph, if you can see, that displayed reduced time or increased motor dysfunction on the rotor rod performance. And then further, we wanted to observe the effect on short-term memory loss. So for that, we did novel object test. And you can observe there was significant decline in cognitive function indicated by the objective recognition index test and the objective object discrimination index, this one. So further, we thought, okay, now we have seen that there is disturbed behavior, like there is impaired motor function, there is impaired memory. We wanted to see if there is any effect on neural inflammation. So for that, we wanted to We use Glasses marker, as Dr. Mehdi has already mentioned about GFAP, that is used as a marker of astrocytes, and that if more and more expression of GFAP basically indicates gliosis, okay? So we observe significantly increased expression of that GFAP in our mutant mouse as compared to wild type and PS19. And to confirm our analysis, or to confirm our result, we did immunohistochemistry while using the same antibody, and I think you can visibly appreciate significantly increased immunostaining in our mutant mouse as compared to PS19 and wild type. Similarly, to observe the neural inflammation, we also did observe the microglial activation while using IBA1 marker, and we observed significantly increased IBA1 expression as well in our mutant mouse as compared to wild type and PS19. And we validated our result by immunohistochemistry as well, and that confirmed our finding with increased immunostaining in mutant mouse as compared to the others. Okay, last but not the least, we wanted to see if there is any effect of that S14A, like proteasome suppression, on synaptic integrity. So what we did, we used pre-synaptic marker, that is synaptotegmin, and we used post-synaptic marker, that is PSD95. So we did Western blot for that, and you can see there was significantly blunted expression of both of these protein in our mutant mice as compared to PS19 and wild type. That confirmed that if there is decreased expression of these protein, it shows that there is synaptic loss. Okay, and then we confirmed our finding by doing double immunostaining while using the same antibodies, and we observed reduced immunolabeling with our mutant mice as compared to PS19 and wild type. So putting all together, we can say that the phosphorylation of RBN6 and by extension, the activation of proteasome by PKA can protect against the proteostasis and can mitigate the pathogenesis in both the brain and heart in the tauopathy mouse model in Alzheimer's disease. So lastly, I would like to acknowledge mainly my mentor. As I told you, this research was part of my postdoc, so Dr. X.J. Wong was my primary mentor, and then I would like to thank my collaborative mentor and then all the lab members that do help me at every aspect of this research, and then the support from other faculties, and finally the funding resources that I was able to do this research. And lastly, I would like to thank you all for listening my presentation, and I would be happy to take any question if you have.
Thank you so much, Dr. Ijaz. I will close this. All right. Thank you.
So do you have any questions?
I think, yeah, I think yeah, exactly, yes. Thank you. Very interesting talk. You talk about cardiotoxicity of proteasome related to proteasome. I think if there are any second-year medical students, we talk about proteasome inhibitors, how they can cause cardiotoxicity. So you can actually see the mechanism, which is very interesting. Next, we have Dr. Ishfaq Bukhari from Kentucky College of Osteopathic Medicine. Dr. Bukhari. Dr. Bukhari, time, 15 minutes. You can move the slides from here. You can use the pointer if you want from here, and make sure that you level the mic.
Sure.
And I'll let you know when 1 minute is left.
Sure, thank you. Thank you, Dr. Ghayur. Thank you so much. Today I just wanted to share my personal experience working through different medical schools and my interaction with students, undergrad and professional colleges. So this is also food for thought for all of you, and just come up with some of the suggestions that you have, and I would love to have some of the questions that you may share what's going on in the medical education, especially looking at the current curriculum trends and that we see in the teaching methodologies that we are using in different medical schools. So as you know, the integration of curriculum is very, very important. It's quite popular these days in most of the medical schools around the world. And more than half of medical schools in the US have already adopted integrated curriculum, and internationally this trend is really tremendously increasing. And the reason is that they want to integrate clinical, basic biomedical sciences with the clinical relevance, and early exposure of clinical training, as many of the regulatory bodies are now focusing. And we also look at the medical education is constantly evolving, introduction of explosion of knowledge. You could see discoveries, and the, as you can see, the role of AI that we discuss is very, very important. And you can see here also the trend that the healthcare is mostly integrated now, integrated healthcare system. And studies curriculum are the care is patient-centered and curriculum is student-centered, right? So if you have integrated care, whereas teamwork is involved in the healthcare service, we need to have integrated approach as well. It's not only factual knowledge, because when students graduate from medical school, when they go in their professional life or medical rotation or residency program, the environment is quite alien to them because they are not actually trained during their professional colleges. The system that they are going through or the educational training is not maybe thoroughly investigated. So as we understand, I'm taking pharmacology because I'm professor of pharmacology. I dislike the boundaries in medical education. I don't like that pharmacology is a boundary. Pharmacology is one of the discipline as an expertise, so that's why I present my example. What do I see working in a traditional curriculum pharmacology, and then how the integrated approach is changing the concept in pharmacology. So why this shift is, as I mentioned, healthcare system is integrated and patient-centered, and we have student-centered curriculum must be needed, and that's very important. And as you could see here, the licensing exams are quite integrated these days. So you have board-style questions. So it's not factual knowledge of pharmacologists, what is the adverse effect? Do I need, as a pharmacologist, to talk about every single adverse effect? And do I expect from you that you should memorize all those things? I still remember my student coming after an interview, you know, with a professor of pharmacology, crying. And I asked her what happened. She said me that she told that a professor asked me, what is the half-life of cephalosporin? So I say, oh my goodness, out of 1 million drugs, how come in his mind cephalosporin? You are lucky that he didn't ask about something else. So this is something important. This is why the shift is there. And we want to initiate because with the recent, as Dr. Praveen already mentioned, the attention span is decreasing. So just 15, 20 minutes in 50-minute lectures, and how much is the retention time? And how much is the consolidation of knowledge? Many clinical literature or many of the studies that we see around the world, not US, around the world, 5 to 10% they get out of lecture. And how much they get from integrated system, interactive kind of a learning, different levels of either case-based, problem-based, or teaching to others, 70 to 80% retention time. So there's one of the reasons that we also need to make lecture interactive, integrated, not teacher-centered. That is the major challenge that we see, and that's why these challenges are basically opportunities. And I see these challenges based on the literature and also my personal experience with many students in different medical schools. And also with the high school and undergrad teaching, because these are not aligned. Each one has got a discipline as a domain. Undergrad is not aligned with the vertical training, like where they will go in the professional college. What would be the teaching methodologies? What are the complexity in different medical education and the training programs, for example? And one of the biggest challenges that I see, you understand, is the integration is is misinterpreted, to be very honest. It's called integration, is a buzzword. It's not a real-time integration, to be honest. This is one of the and people don't accept that. This is one of the things that we should understand, that in the literature, they say integration is a part of medical education, fully integrated curriculum, while if you look at the actual implementation and productivity in the system, in the curriculum delivery, it's just sequencing of lectures from one week to another, or realign some of the topics. So it's not holistic integration, where you split all those different concepts into relevant models. of education. And this mismatch is definitely is going a lot, because if you look at the medical curriculum, problem-based, active learning, case-based, team-based, flipped classrooms, do we see those? I have interviewed many students in different undergrad school, and I have interacted. I asked them, do you have any idea of these terminologies? Have you worked in a small group? Never. Lecture-based, mostly. And that, too, is virtual learning, right? So these are some of the obstacles that we see. These students coming from such background, with such training, definitely are very hard to adopt to this system, to understand what's going on in with so much explosion of knowledge, overwhelming curriculum, and so much complexity in the profession. And that's why they have panic and stress. And we have all these problems because of the, and the second thing is further worsened by non-availability of medical educationists in curriculum transformation. So when we don't have medical educationists in transformation, we rely on regular teaching faculty for curriculum designing. So this is one of the things, because faculty are already overburdened, teaching most of the time, giving their ideas, trying their best, but there are people who are expert. They look at different systems around the world, and they call medical educationists. They design the curriculum, and the faculty input is very important, and then they give us some recommendation and suggestion, which is really very, very important, very, very valuable. And that's why lack of training, lack of medical education is another serious problem in curriculum transformation and integration. So what exactly is current integrated models? I think everybody knows about that, like problem-based learning, TBL, case-based learning. And case-based are either discipline-based that we use. For example, as a pharmacologist, I may talk about, instead of just talking about the nomenclature or mechanisms or just classification of antihypertensive drugs, many books these days are even if you go back like 20 years back, you may not see this clinical approach. So many pharmacology, physiology, and biomedical sciences books are now starting their discussion with a clinical scenario, making more relevance what you're going to talk about. And then there's an interdisciplinary integration is another big challenge that people are working. Still, there are domains that disciplines exist. We see either biochemistry, pharmacology, physiology, anatomy, pathology. Domains are still there in one way or another. Though the curriculum is integrated, but there's heavy influence of subject specialists rather than looking at subject specialist approach rather than physician or student center or professional needs of a student. Assessment strategy is also very, very unique. This is also another challenge that we teach. As we know, the Harden, if you remember, I forgot the medical educationist, he says assessment drives learning. So if you have a really qualified, really aligned assessment, integrated assessment, that would definitely enhance learning and motivate students to focus on the key points and also retention of knowledge, relevance of knowledge, and prepare them for the board exam and stuff. So let's go through some of the discussion. Sorry. If you look at the model here, I'm talking about, let's suppose, loop diuretics in heart failure and the cardiovascular block. So mostly, if you go back 10 years, or maybe some schools, recently like pharmacy schools and some other like nursing schools, they are still lecture-based. So the teaching style is lecture-based and traditional, while integrated, you have case-based discussion, okay? Learning with clinical context. Focus here is factual knowledge, just mechanism, classification. Here is clinical relevance, application, how the patient are presented, how this loop diuretic is helpful. For example, I could talk about mechanism of action. If I tell students they inhibit these sodium-potassium channel co-transporters in the loop of Henle, Dickerson loop of Henle, and then I expect from them that they would remember these concepts in the exam or board exam. So what basically you're asking here, you give a patient scenario with acute failure, how patients are presented, and why from out of 10 diuretics you are choosing furosemide, and how does it help? So here is a good integration of different disciplines like renal physiology, pathophysiology, pathology, and pharmacology. But here, you may see just pharmacology is a domain, which is a discipline-based study. And students are definitely not involved, as I mentioned, not active learning, passive learning. And at the same time, MCQs, mostly factual knowledge, testing, recall questions, right? In here, active participation, and the MCQs are basically integrated because of the clinical reasoning, what kind of presenting symptoms are there, what is the approach, and how this drug can help in this condition. Students definitely will remember that furosemide is one of the best approach in emergency situation if patient is presented with a heart failure, decompensated heart with a fluid overload. So that is really a best approach that students can have. And here is a case. For example, you could see here, this is a case of integrated system where a patient is presented and they already have a lecture, for example, in integrated curriculum, medicine. Faculty will talk about the pathophysiology of heart failure, and they understand that ejection fraction 30% is like highly decompensated heart. So patients are presenting with different bilateral lower extremity like pedal edema, for example, and bilateral crackles. And then there's also increase in blood pressure, and the oxygen saturation is also decreased because of the pulmonary edema. So congestion is there. Which one of the following is the most appropriate initial pharmacology therapy? So pharmacological therapy, these all drugs are very, very important, and they have role in heart failure, right? But do I need to talk about metoprolol or furosemide or spironolactone? So they could understand because of the fluid overload, and you mentioned about the diuretic action, which is a high ceiling, working on loop of Henle, and that's a best drug instead of spironolactone, which may be a maintenance therapy. It's not going to help patient in acute condition. So these are some of the things that probably you can see how they are. And now here's the best approach. Like you understand what are the pathophysiology, what are the clinical indications, how the patient are presented. So if you ask question in this format, students will understand the clinical relevance. That furosemide is really very, very important, and spironolactone is not a choice because it's a RAS system antagonist, which is very important, but a maintenance therapy is not an acute management. And here is one of the best things, like NSAIDs are the most commonly prescribed drugs. I think I still have 2 minutes. Okay, so NSAIDs are the commonly prescribed medicine, and it depends on which block you're teaching. If you're talking musculoskeletal block, for example, you talk about NSAIDs, general analgesic purpose, anti-inflammatory actions, for example. But there are NSAIDs you can split in cardiovascular or GI. For example, instead of memorizing all those information, reverse effects, in musculoskeletal, it is used in prophylaxis of cardiovascular, is causing gastropathy, is effective as an anti-inflammatory, for example. These are all information dumped there in one module. You split it, the cardiovascular system here, to MI. So the low doses of aspirin is very, very important. This is, again, the traditional approach. Oh, I'm sorry, why it's not going? It's locked. Not moving this some Oh yeah, I think this thing is open. Okay, perfect. Yeah. Okay, thank you. And here's also the different models, the AI generated models. What they say, what they look at the traditional teacher-based student-centered activities in didactic. And you could see also the different statistics I mentioned here, 5 to 10%, and 40 to 80% depending on what kind of activities we are using. So this is not only just the space, the ambience itself, the environment, and how the teacher role is just facilitating and self-directed learning where students are working in small groups. And then this is what we talk about the journal. Traditional classical pharmacology lecturer is a pharmacologist if you're doing PhD in pharmacology, not as a physician. So you could see here NSAIDs are working this and this, COX-1 are inhibited, used in prophylaxis, okay? So this approach, I mean, it's not going to consolidate these information. It's not going to impart anything related to use of low doses of aspirin in cardiovascular. But if I integrate the system with the whole physiology and pathophysiology, talking about endothelial system, talking about the platelet segregation, how endothelium functions is impaired in cardiovascular system, how the platelets are activated, what are the transmitters released, ADP, thromboxane, for example, and what happened basically, baby aspirin, low doses, inhibiting COX-1, their thromboxane generation is inhibited, and that's why it has got antiplatelet action. In low doses, you could see here the role of COX-2, for example. They can appreciate the beauty of this presentation because COX-2 is generating prostacyclin that are protective, vasodilatory, and antiplatelet, natural dilator. So it's giving the whole integration, role of aggregation, role of endothelial functions, and the role of different targets. And what if aspirin, people show resistance, how would you combine these drugs? And what is the role of anticoagulant and how they are different from antiplatelet, for example. So all these mechanisms are really important, and then you need to understand the, we call it, the question. Which one is more? Like this is the question, which mechanism, which is quite recall kind of, although there is a clinical relevance, but this is quite a simple question which is not encouraged by medical education is, and the best thing would be, this is the approach. Look at the way how patient is presented, hypertension, smoking history, age of the patient, hyperlipidemia, which may cause atherosclerosis. So they have covered all those pathological concepts, how endothelium impairment would cause these damage and how it will cause aggregation or thrombosis, for example. And these are different drugs the patient is taking. So based on the history, the cardiologist is prescribing a prophylaxis therapy with aspirin. So that's another approach. How would you prevent complication of diseases? So it's give you the holistic approach of aspirin, and it's quite different from what we talk about NSAIDs in the musculoskeletal system. So that is the best thing of cardiovascular system, where you talk about the role of NSAIDs in preventing MI. And that there's a whole bunch of flow chart that I usually use for my students, and the good experience, you can integrate different systems. For example, cyclooxygenase, the whole arachidonic acid pathway, we have leukotrienes. You throw this image to the patient, to the students, and they would come up with the suggestion. What are the leukotrienes pathway? Where exactly they are relevant in different disease conditions? They will say asthma, COPD, for example. What about prostacyclin? We mentioned about low doses of aspirin. They should not be inhibiting prostacyclin generation. And what about the thromboxane, cyclooxygenase 1, which is in the platelets? So you could see different interplay. The biochemistry, the physiology, pharmacology, pathophysiology are coming together, making nice drug targets. So this is really a good drug target, and you can easily predict. So the knowledge that you get is consolidated, okay? And it's retrievable. And it's also very much aligned with the system that we see here. And I think with this, I already mentioned like how you identify different drug targets. You can give, like flipped classroom, you give this image to the students and come prepared. We will talk about different clinical conditions. They will talk about eye, for example. What are the autonomic systems doing to the eye? Oh, in glaucoma. Okay, we are using constrictor. People dilator. Where exactly? In clinics. What do you do with the dilators? We do because eye has got different condition, either by glaucoma or you cause mydriasis to just get the retinal examination. So all these are some of the things that I mentioned, and thank you so much for your attention, and you are most welcome. And I mentioned about this one thing which is really very, very important. Artificial intelligence is a human invention. It takes real intelligence to use it meaningfully. So that is very, very important. So don't rely on this. If you are intelligent, you give good input, you get good output as well. And thank you so much.
Thank you so much, Dr. Bukhari. Dr. Bukhari is going to be around during the poster session, so if anybody has any questions for him, he is very approachable. Okay, so the last talk of this session, we have Dr. Iqbal Hassan from the new dental school. Dr. Hassan, please. So the time is here. You can move the slides up and down.
Okay.
I might indicate you when 2 minutes are left, and then you can adjust the mic.
Sure. Thank you so much. And I'm truly honored to be presenting here among such eminent colleagues. I'm essentially a teacher and a clinician. But I learned from somebody who I look up to, a very well-known researcher in dentistry. I was going to present a case at a symposium in University of Kentucky, and I thought, okay, it's just a case report, why should I present it? And he said, do you know BMJ reserves a section in their journal for challenge cases because they generate clinical studies. So it's important to show unique cases. And this is something, because I've worked so much in hospital, I have come across frequently, but I always felt it goes ignored. So we talk about term zoonosis, and basically this phenomena is where species, parasites or microorganisms jump species, and they come from animals to humans. But the alarming thing is that if you look at this data, 75% of new or re-emerging infections or diseases. So we are seeing increase from 60 to 75%, which is, there is a reason behind it, why we are seeing that increase. And I know we hear about common diseases. When we talk about why, because viruses, they cause acute infections. So we hear about it in news channels, newspapers, everywhere. Bacteria can also cause a lot of acute infections, so we hear about it. Normally, we don't hear so much about parasites and funguses. And that goes ignored. And I have seen some of the worst cases in hospitals that you can imagine. So common zoonotic diseases, and these are the common diseases, so we all have heard about it. Of course, I'm sure nobody here has forgotten COVID-19, so it's still fresh in all our memories. And funguses, we talk about ringworm, blastomycosis. Histoplasmosis is something which you see a lot in hospitals. I've seen many cases. But sometimes the presentations of these diseases can be very unique. And sometimes you'll see funguses which are outside these spectrums. Parasites, we all know about these parasites. So I won't go too deep into it. Just explaining a little bit how this works. So these organisms, it could be viruses, bacteria, or a parasite, or funguses, they first invade the animals. And from animals, it comes to humans. And that's when the problem starts. And that's how we are getting a lot of, if you go back few slides, you're seeing 75% new diseases emerging. So let's talk about fungus. This is something which, and because of my background, I've done hospital dentistry and oral medicine. I got called a lot from the hospital for unusual respiratory distress, unusual kind of infections. And a number of times, it was ruled out, okay, we cannot find out what's going on. And it came out, boiled down to funguses. And my first experience was as a resident. There was a case, and I trained in Charlotte, and nobody could figure out what was going on with this child, 7-year-old child, and he lived on a farm, and they couldn't figure out. Finally, they got the ID doctors, and I know there are a lot of medical students here, so ID people, we need a lot of them. When you work in the hospital, you realize their importance. And the ID physician did something remarkable. He sat down, the child was homeschooled. He sat down with the parents and grandparents and said, we want his routine every single day when he wakes up. So they had rabbits, hens, goats, everything on the farm. But they lived on the hilltop in North Carolina, in Appalachian area of North Carolina. And the child used to, like the granddad used to clean up the rabbit droppings and then throw it down the valley. And on the way back, the child used to get on the wheelbarrow and ride back. So he said, just test him for fungal infections. And that was it. So the fungus jumped the species and came to rabbit. So where are we? What does CDC say? Why we are seeing an increase in fungal infections? So climate change, fungi are changing. Antifungal, I prescribe a lot. And I do get patients where I give multiple doses of clotrimazole or diflucan, and they don't respond. They respond to some degree. It won't go away, or it will bounce back. And then they have to be admitted to the hospital and given IV antifungal like amphotericin B. And that's a challenge because now you have an uphill task about finding somebody who would admit them in the hospital and be willing to do that. So that's a huge challenge. And humans, we like to go camping, fishing, hunting, and we don't pay attention because the funguses are in the soil. If there is animal dropping in the soil, the fungus is there. Of course, animals and environment that we live in. So those are the factors. Another source which a lot of researchers are fearing will happen is, we are talking about transplant, and we are talking about xenogenic organ transplant. So if those organs are not processed very well, they're saying we'll see a new spike in funguses, jumping species, if we use organs from animals to transplant. If they are not, because currently the protocol says test for viruses and bacteria. That's it. Funguses are not tested for. So that's a problem. So this is going to be, and I'm sorry, I should have put in the papers that I am quoting from, but this should have been, this is a prediction that this is going to be a public health issue, not too far from present day, because nobody is paying attention. And towards the end, if I have time, I'll give a very remarkable case apart from today's case that I saw in the hospital just about little over a year ago. Rates are likely to increase further, and it's crucial that aspects of fungal infections is addressed timely. So another big thing is that fungal infection, because it's chronic in nature, because it's not aggressive, when people get fungal infection, the symptoms are very subtle and it gets ignored. And that's the problem that we are having. When it gets ignored, it gets very aggressive after some time. And I've seen that oral fungus do crazy things. I've seen patients go into respiratory arrest after starting with oral fungal infection, which went down. So you see all that, and when you are in hospitals, you see those things. Health education, increasing awareness, and hopefully doing studies on this. So this is a case of a 30-year-old female who's a healthcare provider, and she still reaches out to me. She's in North Carolina. I saw her over there. And she was referred to me after seeing about 10 providers, both medical and dental providers, and rule out any infection of the teeth or anything. And medical providers basically ran several courses of antiviral and antibacterial medications on her. Although I had the x-ray, I still thought, okay, I miss things, I'm human, other people can miss things. I repeated all the x-rays, nothing. And patients' other symptoms were night sweats, loss of appetite, insomnia, brain fog, difficulty trying to explain what she tries to say, joint pain, fatigue, bruising, and rash. Medical history, premenstrual dystrophic disorder, anxiety disorder, ADHD. These were the medications the patient was on. She's married, 2 young children, she's a nurse, and you can imagine she's a healthcare provider herself. So she had access, and she works for a hospital. So she had access to everything that she could. And husband is also a healthcare provider. So everything that is possible that she could do, she was doing, but she was not getting answers. And this is how she looked like when I saw her. And she had lesion near the eyes, lesion extraorally, and then intraorally. I know it's hard to see because she was in pain, but this is where lesion was. So it was right in the line of extraoral lesion. And that's when I thought, okay, we have done everything. And her CBC was negative. And she was also evaluated for rheumatological disease, celiac disease, Lyme disease, which were all ruled out. All her tests I retested, and they were negative. And she was treated with multiple medications. So what was I left with? This was my differential diagnosis: lichen planus, systemic lupus, and fungal infection. And fungal infection was actually at the bottom. And so I called her pharmacy and told them to compound dexamethasone elixir and nystatin mouthwash, and also added clotrimazole troches for 2 weeks, 14 days. So patient reported around 20% improvement, and that was like a light at the end of the tunnel. At that point, I was almost positive that she has fungal infection. She stated that although this is the first time she has seen any improvement, but it's not there yet. Another discussion, and she basically, I asked her more details. So she lives on a farm, and part-time she processes deer, and she also has farm animals. That's when I convinced her to get a biopsy done, and she was very open to it. And it came back, I copied and pasted. Rare fungal hyphae highlighted by PAS fungus stain, no evidence of malignancy. I called the pathologist, he said most likely jump species from farm animal because we don't see funguses like this in humans. And another case like this, and she had to be admitted to the hospital and basically treated with IV amphotericin B and multiple times before she started feeling better. And this is another case which I saw, oral manifestation of histoplasmosis. This was referred as suspected cancer. But one case which I saw just before, I'm good on time, right? Yeah, one case which I saw just before joining UPIKE was at UNC Hospital. There was a patient admitted with severe respiratory distress. And ENT did the eval, and they rule out any ENT-related issues. And my resident was called. She said, and ENT had written in their note, and no criticizing anybody, that they didn't see anything in the mouth. My resident went and she said, Dr. Hassan, I'm just out of dental school, but I think I saw something near the throat. But I could be wrong because the light was low. I go in there and see a black patch towards the oropharynx area. So although they had ruled out fungus, I still went to the nurse. I said, we need to swab, and if needed, we need to get an oral surgeon to do biopsy of that area. And it came out to be fungal infection, which was causing respiratory distress. And again, this guy was working on a farm. He was a farm hand, dealing a lot with not just animal, but soil also. So the point is, we do need to pay attention to these. There are so many medical and dental students sitting here, well, future dental students and current medical students. So paying attention and listening to your patient and trying to understand what they are saying. One thing which I learned, I was very fortunate, I trained with some of the best people here in the country, is your patient gives you the diagnosis if you pay attention and listen to them. Any questions?
All right, everyone. We're coming to the end of our student poster presentation section and getting ready to begin the student oral presentations. Hope you had a chance to make it around to all the posters you were interested in seeing. Everybody's done an excellent job. They all look really, really good. So when we do the student oral presentations, each person will have 10 minutes. And if we have an opportunity at the end, if they have some time, we'll take a question from the audience as well. So we're going to go ahead and get started. I would like to bring up our first student presenter, Christopher Bush from the Kentucky College of Optometry.
Good afternoon. So what we did at my undergrad at St. Vincent College, we looked at on how synthroid negatively influences visual acuity in mice, and we looked at implications for thyroid eye disease on this project. So I just want to give a little background of the thyroid gland and thyroid eye disease. So thyroid eye disease, of course, results from thyroid dysfunction itself, and it can be from hyperthyroidism, which is an overactive thyroid, hypothyroidism, which is an underactive thyroid, or even a euthyroid where there is no signs of hyper or hyperthyroidic effects. And then thyroid eye disease itself has multiple classifications. It can be seen as Graves' disease, which is associated with hyperthyroidism, and this is the most common presentation, and it's characterized by the thyroid-stimulating immunoglobulin, or the TSI antibody. It can also be seen as Hashimoto's thyroiditis, which is associated with hypothyroidism, and it's characterized by the thyroid peroxidase antibody. And then, of course, thyroid eye disease can occur in euthyroid conditions as well. So then a little bit more into the disease progression of Graves', since this is what we tried to look into with this study. So, of course, I said before, it's characterized by the thyroid-stimulating immunoglobulin. There's not much known about this immunoglobulin other than its presence to help signify Graves' disease and possibly some forms of thyroid cancer. So what it does is it acts or mimics like the thyroid-stimulating hormone, or TSH, which comes from the anterior pituitary gland, and this leads to an increase in T4 and T3 production from the thyroid itself. And then, of course, you have the progression to thyroid eye disease, and it's classified by two major phases, the active and the inactive. The active phase is characterized by inflammation of the orbit and the surrounding area, especially in the thyroid itself. And there's also the inactive phase, which is characterized by subsidized inflammation when the inflammation goes down, but there's orbital remodeling, such as fibrotic tissue of the muscles and the fat around there, which keeps them in this, in the proptosis or the bulging eye effect state. So then a couple treatment options for Graves' disease. There's the thyroid-directed ones, which is radioactive iodine therapy, where it destroys cells in the thyroid by shrinking it. It's not first line because, of course, radiation into any kind of body is not really ideal for anybody. Then there's also pharmacotherapy. Methimazole is the first line drug used for whenever somebody has Graves' disease. There's also surgery that can be done as a thyroidectomy, but this is a last resort because, of course, cutting into the thyroid, taking it out is nobody's first option either, so we leave that as a last option resort as best we can. You can also have beta blockers as well that block the effects of the T4 and T3 hormone. Granted, it does not stop the thyroid from producing those hormones. Then there's also treatment options for thyroid eye disease in itself. You can use corticosteroids to help with inflammation. Tepezza is a new form of therapy for insulin growth factor 1. It targets the receptors for that and stops the thyroid eye disease from occurring. Prism can be used to help with any kind of double vision that the patients have, but it's more of a temporary effect. Orbital decompression surgery is also a last line resort when there's too much inflammation, too much fibrosis, where the patient is at risk for optic nerve compression, and there needs to be surgical intervention immediately to keep that from happening. Orbital radiotherapy can also be done, but that is also not very well understood in how it affects thyroid eye disease. We just know that it can help with it. So then the hypothesis for this study is that the treatment of mice with synthroid or levothyroxine, which is also what it's known as, will show a significant reduction in visual acuity via mimicking Graves' disease, including thyroid eye disease symptoms. So for our methods, we had 15 C57BL6 mice randomized into 3 groups. Our first group was our control group, where there was no treatment given to the mice. Then we had 5 mice in what we deemed a low dose group of human equivalent dose of 50 micrograms of Synthroid, which was about 4 nanograms per milliliter in their water. And then the last 5 mice were in what we deemed a high dose group of a human equivalent of 100 micrograms of Synthroid, which was about 8 nanograms per milliliter in their drinking water. Then we did visual acuity testing with a custom-built optodrome that we based off a zebrafish model. And this assessed the optomotor, or the head movements, and to a lesser extent, the optokinetic eye movements of the mice. And the rotations consisted of 2 counterclockwise rotations and 1 clockwise rotation, each rotation lasting 45 seconds. So then they were assessed for 8 weeks on body weight, the visual acuity, food consumed, and water consumed. And the visual acuity, we assessed the rotation for the visual acuity when it lasted for at least 2 seconds, and I have a video here I'm hoping it'll play. There we go. You can see right here that the mice is already starting to get used to the rotation there. And right there is a small rotation as it starts to go. And then there's a bigger rotation right there that lasted for about 2 to 3 seconds. So that would count as a rotation for that mouse. So then for the results, for the body weight progression, there wasn't much difference between any of the groups, and this is just to ensure that the mice were not significantly gaining or losing any weight. As you can see, there was a trend upwards, but it was pretty consistent across both of the groups, or all of the groups. And then for food consumption, this is to ensure that the mice were consuming around the same amount of food. There were a couple jumps in weeks 2 to 8, but overall it stayed pretty consistent across all the groups. And then for water consumption, this was just to ensure that the mice were consuming the water, since the dose groups had Synthroid in their water. As you can see, that it was all pretty consistent across all the groups here as well. And then for the change in counterclockwise visual acuity, I'm going to pull out the laser pointer here. So we use a p-value of less than 0.05 for as a significant value here. And as you can see, in weeks 7 and 8, there was a statistical difference between the control groups and both experimental groups at those weeks. And then for the clockwise visual acuity, there wasn't as much statistical difference here, because since they only had one clockwise rotation, that it probably accounted for more error that occurred, which led to bigger error bars like in week 3 right here. But there was still some statistical difference between weeks 4, 6, and 8. And hair loss was also exhibited between the experimental groups, which is, again, a known side effect of Synthroid. You can see that there's some major patches of hair thinning and hair loss on both of the groups, and this right here is the 4 nanogram per milliliter group, and this right here is the 8 nanogram per milliliter group. So what can we conclude about this? The Synthroid administration led to visual acuity reduction in the mice, which possibly modeled these TED-like symptoms seen in Graves' disease. However, these findings do underscore the need for further investigation into Synthroid's possible ocular effects in humans, and we need to focus on the mechanisms driving these changes and optimizing therapeutic strategies for patients with thyroid eye disease. So a clinical message that can be taken away from this as well is that periodic ocular examination, including visual acuity assessments, should be considered in patients with thyroid eye disease for early detection on any drug influence or any physiological influence on vision. Specifically, you'd want to look at those taking Synthroid for Hashimoto's and for any kind of underacting thyroid, basically hypothyroidism, to see if they flip from a hypothyroidic state into a hyperthyroidic state, since Graves' disease and hyperthyroidism is a more common presentation of thyroid eye disease. You would want to watch for that. And is there any questions?
Thank you very much, Christopher. All right, so our next presentation will be from Disha Patel from the Kentucky College of Osteopathic Medicine. Disha, we'll get your presentation up here. We'll start here in a second, yeah. All right, we'll get it started, and then he'll count up in the corner there for you.
The what?
As soon as it starts to present, he'll start to count up for you, so you can watch your 10 minutes.
Hi everyone, my name is Disha Patel. I'm a second year at ChiCom, and for about a year now I've been working with Dr. Hahn in the research lab studying the interplay of ear alpha, ear beta, and GPER receptors with isometric tension studies on porcine coronary arteries. Okay, so cardiovascular disease is the #1 killer in America. Estrogen plays a vital role in that in women's health. When women get older, their amount of estrogen decreases. Estrogen plays a protective mechanism. It causes nitric oxide release, which vasodilates our arteries. As we get older, we lose this protective mechanism, and that's what causes our arteries to stiffen, and there's nothing that really helps us not put us in that risk for developing cardiovascular disease. If you look at the graph, you can see that older women are at a much higher risk of developing cardiovascular disease, and unfortunately, there aren't that many therapies that can help change that difference. Women's Health Organization actually released research that supported estrogen being a negative as a hormone replacement therapy method because of the data that they studied. What we hypothesize is that the mechanism is not fully understood. So we know what estrogen does on ER alpha and ER beta and how it signals enos and nitric oxide release, but what we don't know a lot about today is GPER. There's a lot of things that are hypothesized, but I won't go into the details of what we do know. I'm just going to simply tell you what we're seeing in lab. So these are some of the experimental designs that we use to study different avenues based on our hypothesis. We used sRNA transfections. We silenced certain genes to see if we can study each mechanism specifically. We did tension studies, and this is basically mounting arteries onto these machines that can help measure the amount of contraction or relaxation the arteries are going, or that's happening in the arteries. And that way we can see whatever we're treating the arteries with, the effect can be compared on a statistical analysis basis to see if there's different treatments that cause more relaxation versus less. So it'll make more sense when I show you this. What we hypothesize is that there's a huge complex that works together. Estrogen will recruit ER alpha and ER beta to work alongside with GPER and the GI subunit to collectively activate enos and cause coronary artery relaxation. With the graphs that I'm going to show you, we're going to target each one of those complexes to see the effect it has on enos and the coronary arteries respectively. So we'll target GPER, we'll target ER alpha, and we'll target ER beta. And you can see the importance of looking at the whole complex instead of just one part of it. All right, so this graph shows us what we saw when we just treated the arteries with estrogen, which is this orange line, and then what happens when we use G36, and that targets GPER to amp up the strength of what it's doing. What we saw was that there was a significant increase in nitric oxide release, and that's causing more relaxation on those arteries. Over here, the orange is estrogen alone, and then the pink is with ICI. ICI is an inhibitor of estrogen alpha receptors and beta receptors. What we saw was, yes, there's less relaxation, but there's still significant relaxation. This suggests to us that there are other receptors in play that people are not considering as much as they should. Over here, we took it one step further and we did our sRNA transfections. The previous two charts that you saw were just using pharmaceutical therapies of agonizing certain receptors. This is actually gene silencing them. And when we silenced GPER, we saw a significant decrease in the capability that arteries have to relax on its own, with just relying on ER alpha and ER beta. This was a huge suggestive factor that GPER plays an essential role in the whole relaxation mechanism. This is a summary of the findings that we once had, just going over everything that we just talked about. E2 causes arterial relaxation. G36 in conjunction with GPER will increase that. ICI will reduce that, and GPER knockout also reduces it. New data that we've been doing recently was also knocking out ER alpha and ER beta to see how strong GPER is by itself. What we see is that, yes, the capability of relaxation is a lot less without GPER, or without ER alpha and ER beta, but it's the same thing. Altogether, this just shows us that all three receptors are crucial in the signaling pathway, and they work together. On the other side, I just provided this picture to show bradykinin is a target that we know about today that causes the same relaxation, and it's really interesting to show what estrogen does in comparison with bradykinin, something that we know that relaxes arteries. In the future, we are going to continue our Western blots. We have taken a different perspective on things to experiment the same understanding that we have gained through isometric tension studies and look at them not just on an artery basis, but on a cell basis, do the same treatments to endothelial cells and see if we can show the same mechanisms we're seeing on isometric tension studies in Western blot analyses. And what we've been seeing is truly exciting because it's supporting some of those hypotheses that we're making. And then we want to do nitric oxide releases, so that's in store, and we're super excited. At the end, we also just hope that we're building foundation for the future and future drug therapies for women's cardiovascular health. Thank you. And these are people I want to thank. Oh, yes, yes. Questions? Okay, thank you.
All right, thank you very much. Our next presenter will be Kunj Vyas from the Kentucky College of Osteopathic Medicine. Oh, you're good. Guess I'm going to start the presentation down here, and then you'll see that it'll count up your time in the corner here.
Okay.
There we go.
All right. Hello, everyone. My name is Kunj Vyas, and so today I'll be presenting a study about the popliteal injury and its relevance to knee dislocation, looking at things like anatomy, diagnosis, treatment, and outcomes. And so these are the authors that were able to work with me for the past couple of months on this paper. So why is the popliteal artery and its relevance to knee dislocation important? So misdiagnosis is critical, avoiding that, because it can lead to ischemia, necrosis, and amputation within hours. So functional outcomes remain poor even months to years later despite advances. And we need a comprehensive bridge between vascular and orthopedic perspectives. So the study objectives here were to review the anatomy and the mechanisms of injury, summarize any diagnostic challenges, and look at the different various modalities, analyze and synthesize the current functional outcomes. So in terms of evaluating the surgical techniques and highlighting any recommendations for future research. So looking at the anatomy, the popliteal artery is a continuation of the superficial femoral artery. It's tethered proximately with the adductor hiatus and distally with the soleal arch. So it's kind of the fixed position that makes it very vulnerable in high energy trauma. Although we should note that in high BMI patients, even low velocity injuries, such as stepping down from a curb or sitting up from a seated position, that can lead to dislocation. And typically posterior dislocations, they're the ones that are going to present with the highest rate of arterial transection. So this is kind of a depiction of the course of the anatomy of the vessel. And there are different anatomical variants, such as the common bifurcation that's seen here, but there are also trifurcations, so 3 kind of branches off that artery. And there can also be high divisions where this artery is actually much higher than in the typical person, and that appears in 4% of people. So this is surgically relevant in terms of imaging and ensuring that the vascular surgeons are aware of this, because with different mechanisms of injury, this can impact the actual repair. And there are some different classification systems. So the Kennedy system, which is a direction-based, whether it's anterior or posterior. And then there's the Schenck classification. So this is ligament-based. The number of actual ligaments that are injured, such as the MCL, LCL, ACL, PCL. So the Schenck classification is currently the most used one all throughout. And so the presentation and diagnosis is sort of like this interdisciplinary approach of first responders, emergency physicians and nurses, and then the orthopedic and vascular teams. So the clinical presentation comes in with pain, pulselessness, and distal ischemia, and they may have a normal exam due to the collaterals, the other vessels that are in the knee. And so, as I mentioned before, those high BMI patients, they may present atypically. And so looking at the diagnostics, one of the things we looked at, one of the most valuable tools is the ABI. So this is comparing the antebrachial, like the arm blood pressure, with that of the ankle. And when comparing that, those two and creating a ratio, if it's less than 0.9, that's indicative of distal ischemia, so ischemia in the legs. So this is a brief summary of the different diagnostic modalities. And as you can see, the ABI is very valuable because it has 100% sensitivity and specificity. Currently, the gold standard is the CT angiography, though. And things like physical examination with a physician or a nurse or ultrasound, they can be operator dependent and lead to misdiagnosis, especially if those collaterals are providing additional blood flow. And this is just a brief management algorithm. So with that initial trauma presentation, looking at the different pulses, whether it's asymmetric in the legs or symmetric, and how the ABI impacts what is the next route and next additional step for the patient. We also want to look at the long-term functional outcomes in terms of return to activity. So we did a Prisma-guided systematic review of those functional outcomes using PubMed. So we started with initially 144 studies, and after full analysis of these papers, we ended up with 7 studies. And the inclusion criteria was cases with popliteal artery injury and looking at long-term functional outcomes. So these utilized various different scorings. So there's the Lisholm score, the IKDC, which is like International Knee Score, Hospital for Special Surgery, and the Oxford Knee Score. So basically, after coming through with all these different studies, we found that there's a lot of heterogeneity in the way this is analyzed. And so we use a risk of bias assessment called the Modified Monitors tool to assess for any risk of bias. And so this is a brief Prisma diagram of working through those different papers and how we reached our final 7 papers, excuse me. And so in terms of study characteristics, all 7 studies were retrospective in nature, published between 2008 and 2020. And the sample sizes were relatively small, ranging from 11 to 125, and the ages ranged from 30 to 48. And one of the inclusion criteria that we had was specifically looking for follow-up of greater than or equal to 18 months. This ensured that we're looking at long-term and not short-term outcomes. And typically all throughout, the vascular injury management was pretty standard, looking at bypass grafting, such as a reverse saphenous vein graft. So basically just harvesting the saphenous vein and utilizing it, connecting where the actual popliteal artery was disrupted. These essentially highlighted all the studies that BMI, age, and surgical timing are key outcome modifiers. And overall, using that risk of bias assessment tool, the comparative studies scored between 14 to 17 out of 20, and the non-comparative study was 9 out of 12. So overall, these were strong studies, but they had different weaknesses due to the nature of the injury and kind of the rarity of it and the small sample sizes, and them being retrospective in nature as opposed to prospective. So our results show that across all 7 studies, vascular injury was linked to worse function, and these were using different scores, so the Lisholm and International Knee score. And we looked at different comparison groups, so things like age, so less than 30 or greater than 30, as well as with vascular injury or without. And across the board, all throughout, being above 30 and having vascular injury really reduced those long-term functional outcomes. Another aspect that we looked at is reconstruction versus no reconstruction. So a patient may opt to not have reconstruction of the ligaments in the knee if there's extensive vascular damage or ischemia, or if they have significant comorbidities that disallow them to undergo surgery. And looking at the timing of reconstruction, so this Levi study that we included in our paper, it suggested that delayed or absent ligament repair in older patients would result in the poor function. But this will lead to a little bit of controversy and argument that I'll mention in a couple of slides. So in terms of orthopedic considerations, the initial priorities are obviously to reduce that knee dislocation and minimize any neurovascular compromise. So anything that's irreducible will require any specific urgent open reduction. And in terms of imaging strategy, just as I mentioned before, multimodal imaging approach. MRAs are interesting, multi-resonance angiography, because they can also provide soft tissue detail for further operative planning. And in terms of fixation, we have external fixation. So that's typically when the fracture is grossly unstable or there's polytrauma, but there's always the risk that you can lead to arthrofibrosis. So this is going to require recurrent manipulation under anesthesia throughout the patient's recovery. But going back to kind of that controversy I was talking about, especially in German literature, is that something that wasn't analyzed by the studies that we looked at, but this bone first versus vessel first approach. So the bone first approach is going to prioritize the external fixation to stabilize that joint before vascular repair. And it's going to minimize the risk of graft injury during reduction. So when you're reducing and you accidentally nick that artery due to low patency, kind of the whole revascularization of the limb is gone as a result. Then we have the vessel first approach. That's going to prioritize that early revascularization with something like temporary shunting. And that's preferred when you're reaching that 6 to 8 hour ischemic threshold in patients. And then another consideration is the reconstructive timing. So whether you want to do this acutely, staged, or delayed. And so we see in previous literature and kind of the rule of thumb was to do it acutely because this is when the tissue has the best quality. However, we've seen kind of a move towards a staged approach where the medial and lateral collateral ligaments are repaired first and the cruciates are delayed until the actual range of motion is restored. However, at this time, there's no validity to or valid studies that are actually comparing these and studying them and long-term outcomes. And in terms of vascular considerations, so these are the different types of grafts that can be used. So temporary shunt replacement, that helps with initial ischemia presentation. And then there's definitive options. So RSVG is kind of the gold standard that provides the best patency of the graft. And endovascular repair is typically reserved for isolated intimal injuries. So looking at the conclusion and future research, so the popliteal artery injury following knee dislocation, it's limb threatening, it demands early recognition, and there are great things in place, but they need to be kind of organized and properly managed by algorithm. So functional outcomes are consistently poor in patients with vascular injury, especially when the repair is delayed. Reconstruction improves long-term knee function, but return to pre-injury levels remain poor and patient and mechanism dependent. So BMI greater than 30, especially those patients who have a low velocity injury, as I mentioned, standing up or stepping down from a curb, and patients above 30. Those are the risk factors for worse outcomes. And so the limitations of our study. So there was a predominance of retrospective study designs. There were small sample sizes, a lot of heterogeneity all throughout, and there's limited data on the actual timing of reconstruction and the role of nerve injury. So that's kind of what our future research needs are, a prospective study that directly compares repair versus reconstruction timing, whether it's acute or a staged approach, and the actual type of vascular graft that's used, so whether that's a reverse saphenous vein graft or an in situ graft. And so also investigating nerve injury impact and rehab protocols such as physical therapy, occupational therapy, and overall looking at the long-term return to sports. So yeah, that's all from me. Thank you very much. Questions? Or?
Do you have a question? Is this something you're putting together a manuscript on? Right, our next presentation, we actually have two presenters, Evan Wheeling and Andrew Castle, both from the College of Arts and Sciences. All right, and being that they have two presenters going at the same time, they'll actually have 20 minutes. All right, so as soon as we start it, you'll...
Is that the most recent copy of it?
It's the one that's in the folder.
So I'm gonna have to, I did an update. Yeah, we did updates to it. Do you mind if I go to his email real quick?
You can. Yeah. But you might have to go in a different, did you send them one?
Yeah. Oh, he needs to sign it. Can you sign it real quick? Yeah.
What they have? Okay.
I think it's, there it is.
Yeah, do you want to save it? All right.
Yeah, I made some
Yeah,
changes.
it's good.
to it. I think this is good. Do I just download it real quick?
Yeah, you can file, make a copy from file.
You go ahead. There you go.
Create a copy, download a copy. Okay, yes, should be good.
Perfect.
All right, and I think just in case. What is this?
I don't know why it pops.
Yeah, you know what? It just pops. Yeah.
It did ours too. It
Yeah.
did it for ours too. For
All right, so you can just do presentation view.
Okay, cool.
Yeah.
As soon as it starts, you see it count up in the corner here for you.
Yeah.
Oh, perfect.
So that's a count.
All right. I'm gonna take this out. If you don't mind. I don't know if it'll come out. All right, everyone. My name is Ivan Wheeling. My partner here, Andrew Castle. We're going to be doing, we did basically a pH-indicated film, and we did an AI-optimized approach with our mentor, Dr. Hess. So we used AI to kind of give us ideas and help us generate research. So the challenge that we're trying to face, I'm sorry, it's not as bright as I wanted it to be, but I can tell you some statistics about it. So right now in the United States, approximately 2.55% of all U.S. retail beef is discarded annually due to discoloration, even though it's good. So that just visual cues, not having a bright red meat, shows the fact that it doesn't really look good to us and we don't want to eat it. So total waste accumulates to 194.7 million kilograms. That's a lot of beef. Approximately 780,000 unsacrificed animals for them just to be thrown away, and the economic impact of $3.73 billion per year. And the direct losses just from discarded beef is $2.55 billion annually. We also have the idea that this pH film will help us give an actual scientific cue of pH change from protein degradation within the meat. So by just reducing 1% with the pH-sensitive packaging, we save about 23.95 billion liters of water, 268.88 billion millijoules of energy, and reduce approximately 400,000 tons of CO2 emissions. So we all go into grocery stores, whatever looks better, we usually buy it. So these assessments on visual cues when it comes to beef are the issue that we are looking at. Superficial discoloration doesn't always mean spoilage. Some cuts are different than others. Fresh meat is sometimes wasted due to appearance, and we need a more reliable metric. We need something better to go off of. So that's what we looked at. We kind of sat in a room with Dr. Hess, and we had 10 articles, both all discovering something that can go about this, but we wanted to go about a different route. 100% food safe, biodegradable. We wanted it to be basically a film that is reactive, it can biodegrade, it's 100% food safe, you could eat it. So basically we thought about a film. Now it is anthocyanin, and that is from red cabbage extract. There's also a lot of other fruits and vegetables that have anthocyanins in them, but it provides an objective real-time indicator of freshness. So we kind of have to talk about what the AI's kind of input, because that's a part of our research. So the initial challenge we gave the AI is designing a film that visibly responds to real meat spoilage. We went with pH because ammonia gets created based off of recent studies. Ammonia gets created, which changes the pH of the film. Now, the requirement was build formulations that change color with the sink and the meat quality degradation. Their turning point, we introduced AI to a guide. So I gave it a guide. This is, Dr. Praveen talked about it earlier. We call it prompt engineering. You'll be seeing it in the slide later. Prompt engineering is very important to tell how the AI to work, the output, what it's looking at with the input, and giving it a specific role in your research. Now, the results we're going to see is a faster, smarter, and a better creation of a response of a spoilage indicating film. So right now we said, why use AI? Well, it's new. Everyone's like, what can it do for us? What are the actual uses besides students cheating on stuff, right? So traditional research methods are time intensive and they take a lot of background. Basically, this makes people who don't have PhDs like Drew and I, people who have PhDs with the scoutship of Dr. Hess here, we did a need for speed kind of idea with this. We didn't have as much time as we usually would. We had 3 months to kind of come up with a crazy idea like this. And we see tech meets science. A lot of people try to include, Wolfram Alpha was the first one to do it with mathematics, right? They created something that allows multiple systems to ran big equations that used to take a lot of time. Now they don't have to, they take barely any time. And the objective was to use AI not just as a tool, but as a collaborator. We didn't just want to use it as an object. We wanted to use it, and we wanted to keep it a part of our research. We wanted to keep it in the loop. So we used ChatGPT, Claude, DeepSeek, OpenAI API, which you're going to see a little toy I made later with that. TensorFlow, PyTorch, and Hugging Face. So those last three that I said, TensorFlow, PyTorch, and Hugging Face, are all to train an AI. A lot more advanced than me, unfortunately. I had some issues trying to train an AI, so I ended up using a pre-made one, but then teaching it kind of what we wanted to do. We used literature support, so we had to actually gather and synthesize information originally when we first sat down in that office and we talked about what can we do with this AI. Then we had to do a data analysis. I actually did all our titration calculations as well as see any issues that we saw with titrations, running over trial analysis. We did experimental design. We generated new formulations, actually created a negative feedback loop where Drew reports to me about what's the issue with the film. Then I coded the AI, I consult the AI, and then it gives a new idea, generating a faster workflow. And that's basically what I just talked about for that last one. So, and some studies that, I can't really read it, I forgot what. Studies that indicate AI applications can reduce drug discovery times by 50% in clinical studies with pharmaceuticals. Now you're saying with pharmaceuticals. We're going to apply it here because it has relevance due to the fact it's in something new being created. Additionally, AI is shown to compress clinical trials by 6 months due to productivity. So we're going to talk a little about this prompt engineering. I'm going to scan through this very quickly, and I'm sorry you guys can't see it. I expected this to be brighter. So we talked about prompt engineering. Dr. Praveen talked about it earlier. We talk about so if I just ask you, what is a good pH that's safe for food? It's gonna give you something that I don't really want. But if I say, I need a natural 100% pH that's reactive between pH 3 to 8, blah, 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 you get the point. It basically gives the response that we want it to give. And when we give it a structured outline and I understand how it thinks and what I want the output to give me, it gives me a better feedback to put into our loop. Now the convergence. Merging AI and research was actually a little bit harder than I wanted it to be. Creating multiple different chats, AIs have context windows. It's like the amount of time they want to give to a certain thought. We integrated DeepSeek v3, which is a Chinese integrated AI, and O3 Mini High, which is a reasoning bot. And basically this overcame physical formulation constraints such as solubility, thickness, drying times. Machine learning suggested some other different things that we could do. And empirical trials validated AI-guided changes in film pH studies. So when we were looking at it, our first recipe was completely pH sensitive, meaning that it had 100% validity. Now it did use that from previous studies that we gave it. But it was basically, like Dr. Hess likes to say, a big file cabinet that has the ability to put things together that humans won't. So now we talk about examples of convergence. We incorporated 10 peer-reviewed research papers representing diverse set of methodologies and findings. We incorporated these through PubMed and some other sources. Through prompt engineering, like I talked about, we input instructions into physical testing. I didn't put a prompt in here because it would take probably 3 slides to put it in there, but you give it a large amount of data and you tell it the structure that you want. Now we talk about lessons learned. AI strengths, it had rapid data synthesis, high volume pattern recognition, scalable model generation, which basically means we can add a lot into it. And we could actually process over 100,000 words of technical input with almost 100% accuracy. It identified trends, common formulations, key experimental parameters across multiple studies. The limitations we ran into a lot, it didn't understand sometimes a certain unit, or didn't understand what I wanted. I have to go in super depth. When I've created the first synthesis with it, it actually went into wound care. So it didn't have anything to do with what we wanted to do. So now I'm gonna hand you over to my colleague, Andrew Castle.
So now let's talk about what we actually did. In our short tenure of 3 months, we formulated 4 recipes. Now our first 3 suffered a lot from film thickness, they were brittle, they just weren't overall very good. So now we, through the help of Ivan and his AI genius, we developed this new formula, which you can see on the projector screens, of 1/2 gram worth of sodium alginate, cornstarch, gelatin, and a solvent of 26 milliliters of water, tween 80, also known as polysorbate 80, and 4 mils of glycerol, and a very light spritzing of calcium chloride at a 2% solution after approximate 12 hours of worth of drying. What does that mean? Like, what is going on? During the heating process and the formulation of these films, the gelatin protein exposes your hydroxy, amine, and carboxy groups, and these groups formulate the initial network of the film. And now the starch, whenever it heats, it swells and releases amylose, amylopectin, and these help stabilize the polymer network. Now the anthocyanin I will get to in a second. The alginate bonds to the calcium, supporting the film's overall tensile strength. And now the glycerol forms the polymer chains, and it disrupts bonds, and it acts as a plasticizer. So here is our, this is for our research standards. You can find literally all of these are at Walmart or on Amazon. So it is completely 100% food safe. Now, the flavillium ion, it is the backbone of the anthocyanins, which the flavillium ion, as you can see in the top right, is a tricyclic compound, and it has a lot of resonance stabilized positions, and you can see that these hydroxy and carboxy and all these other groups are formed through protonation, deprotonation, tautomerization, and polymerization, and all these can form different colored dyes. Here is our first titration model. This is with a HCl and is about a 0.07 molar. And you can see that this provides us with a sharp pH curve and we can see that we observed the color changes at roughly the pH of 6 to a very light blue, and then pH of 8 to a very light green and above. Then we decided to test it out with a weak acid. We did this with ascorbic acid to see the sensitivity of this indicator, to see if it would give us similar results. And we did. And this is with preservative. And here you can see that all of these pH titrations show very similar equivalence points. Here's your
All right, we're gonna skim through this this IR spectroscopy. So IR basically is a non-destructive analytical technique that measures the absorption of IR radiation by molecular vibrations, basically seeing all the functional groups that are within this film, this anthocyanin. Its role in film analysis and ultimately anthocyanin analysis is the IR spectroscopy is used to really analyze the polymer films and their chemical compositions, detect the certain groups. We see structural insights such as OH groups, NH groups, carboxy groups, ketone groups, and other groups that are in it. We used a Perkin-Elmer, which is provided by the College of Arts and Sciences, and we had specifications based off what was given. So the first one we see is the anthocyanin, and this is dried on Teflon. So this is just dried on anthocyanin, evaporated, there's no liquid, it's just dried anthocyanin. We see a large OH peak, and we see it depicts a conjugated CH peaks and depicts a C double bonded O peak, so carbonyl, and it also shows aromatic, which means it does line up with the chemical composition from the textbook, the raw anthocyanin, it does match it. Now what we said, and here is the textbook one, as you can see right there on my left, it may not look similar in that fingerprint region, which is that last part of that graph. Less unimportant, we are really focusing on the carbonyl. The only difference is that C double bonded O is a little bit not as deep on the left one, which is that peak right around 2930 wave number. So when we're looking at this, we're basically seeing our film is pretty close to the purified anthocyanin, which is nice because they used a lot more expensive equipment than we did. We also talk about the the blank film. So this is the film that he described, my partner here described all the materials, the glycerol, tween 80, all of it in it. So when you look at this, you see OH peak, which is, we have glycerol on it, and we see conjugated CHs, and we see depicts aromatic and standard CC peaks, which is normal with all the stuff we've given. But we compare it, we say, okay, what if we put the anthocyanin in the blank film? We see absolutely no change. We didn't want to stack them because you can't see the change, so we wanted to sit them right next to each other. If you scan through them, there's no difference at all, which shows that the fact that the anthocyanin isn't messing with the molecular composition of the film itself. So then we said, okay, let's do a transmission so if we can see a little bit more in depth. We also see nothing. It looks pretty close. There's just a little bit in transmittance, and that is, what? Yeah, it's just also, it was too thick with the transmission, and that's why you only see certain fields. And we would try to do a Raman spectroscopy, but we did not have the equipment to do that. I'm sure that would have been also helpful. I'm gonna hand you back to my partner for this UV Vis.
So for our UV Vis portion of this experiment, we used the Agilent Carry 3500 UV Vis spectrometer. We used matched quartz cuvettes. We did a 1 to 50 dilution of our concentrated pure anthocyanin solution. And then we did 2 other samples we added ammonia to see the blue color change, as well as the last sample, I added enough ammonia to see the green change. And as you can see, we see a absorbance spectra between the wavelength of 550 and 650. That's the big peak right there in the center of the graph. And then we also see another one that is 480 and off the chart. This is basically the numerical values of what you're truly seeing in person. This shows that through the addition of ammonia and the base, you see the color change. And so what does this film actually look like? As you can see, me and my colleague are holding up a large sheet of this stuff. And of course, we're kind of working on transparency. There is still a few kinks. Homogenizer would be pretty nice. But I would like, on our behalf, to say our appreciation to the CUSP panel for allowing us to truly do this research and allowed for an AI-optimized pH-sensitive film.
All right, so there's our references, but we have like a little portion we want to do. So I spent a little bit of time. Let me get this in here. Crap. I spent a little bit of time making an AI that knows all of our research. So I basically put all the data we encapsulated during this 3-month stretch into an AI, and you can ask it physically any question about our research, and it'll give a response based off the question. So we can verbally ask questions. I created a little pipeline that allows you to verbally talk to our AI. So we'll open that up. And I know it's been quite quiet in here, but if some people could ask some questions so we could test out functionality of this, I would like that, if possible. Let me pull this up real quick. I don't know. I physically can't. Oh, there it goes. That's where you are. Come up here real quick. I don't know where to put it because it's like connected to that.
Oh, okay.
Yeah.
So it wouldn't, sometimes if you drag it.
Yeah, this might be a him question.
Yes, exactly.
I'll tell you the website. Here, it's right here. There you go.
You can go to it. I'm just going to duplicate the screen.
Okay, that's fine.
That way they can see it.
Yeah. Yeah, if you can just. Yeah, I know it's being picky. I think there's too much open. You can close everything, or close it with this.
Where the PowerPoint's up.
Yeah, you can close ours. Yeah.
Maybe.
That's what I'm saying. This thing's being super slow. You gotta give it a second to think about closing it. Yeah. This is so fun. Oh, you got something. There it goes. All right, just open that big one right there. Hopefully it doesn't, no, not, yeah, you can close that one. Go back to the Google. Yeah, this one right here. There you go. Full screen that. All right. All right, here's the website. Hopefully we don't get any technical issues while making this. I did make this in 4 hours and then had to do a little bit more tweaks on it. So basically I bought a $2 domain name, UPIKE Meat Research. I thought it was kind of catchy. All right, so let's just, you can ask any, it could be the simplest question on earth. Go ahead, Wally.
So you guys talked about doing it on meat and steak. Is there any other foods or vegetables, or fruits or vegetables, or any other kind of food that you guys could use the film on and test for the, test for what you guys are testing on it?
Okay, so I'll ask that question too. I'll kind of simplify it. So is there any other foods besides just meat that we could test this on? I mean, not just meat in general, but chicken or anything else that this could be tested on. So unfortunately, I don't have a $100,000 server, so it takes about 15 seconds to generate a response and scan through all of our results. I was told the sound will play above, so hopefully it's not so loud that everyone just absolutely gets killed with it, but we do also get on screen. Okay, also, I'm gonna add this on top. You can also ask this thing a calculus question or anything else of like any AI matter because it is an AI. It just uses our background knowledge we've given it as the first line of defense. If it's not in that, then it will go to searching its actual neural system. If it's not in that, then it will actually search the internet. So any other questions? Yes. Do you want me to ask the question? Okay. If I have food in the fridge for 1 week and I leave it in the fridge, can I eat it after that 1 week? Yeah, that's a good question. See if it actually works. I wonder how it's going to respond to that though.
It depends on the type of food and how well it's been stored. Generally, leftovers are safe for about 34 days in the fridge. For a week, using a pH-sensitive film, like the one Ivan and Andrew are developing, could help you determine if it's...
Oh, it even mentioned us. All right, come on, a couple more questions. No. This is completely, so I had to do a little bit of a study on it just to make sure I could input data. So the way they treat it is ChatGPT is considered open source. DeepSeek is considered open source. So that means they are technically free at the lowest level. Now they allow you to do paid tiers, right? When you actually, ChatGPT has its smallest model. So once it steps up from 4 to 5, then 4 goes free on a certain network where you can actually train it yourself and give data research or any type of anything you want to put in it in its own category. Now, like people were talking about, is it your intellectual property? Everything that ChatGPT generated is Andrew, Dr. Hess's, and my intellectual property. I've looked through multiple research papers, APA citations. We actually cited ChatGPT in the references because it has to be cited, but it's considered a transformer. So basically it's like a Grammarly on steroids, but it allows you to generate content based off what it's given, since it's pre-trained by humans. Okay. I can ask that question, and I'll ask the AI that I can answer that question. I'll let the AI also answer it. Yes, it's a physical film. It's not permeable at the moment, so nothing can go through it. We can let water sit on it. We can let milk sit on it, and it just acts like Saran wrap almost. It's basically a Saran wrap with a pH-sensitive film in it. But I will ask the same question to the AI. Technically, if I spill milk on this film, will it go through the film? Will it cross-contaminate? So let's see how it responds. It's not going to, it's a film. That's a good question, though.
The film is designed to be a barrier, so it shouldn't allow liquids like milk to pass through and cause cross-contamination. The materials used, like sodium alginate and gelatin, form a stable matrix that traps the pH-sensitive indicators and prevents leaching. This makes the film effective in providing a visual indicator without compromising the food's safety.
There's your question answered. All right, thank you guys so much.
Thank you, Andrew. Thank you, Ivan. I'd like to play around with the AI just a little bit more. It's fascinating. just one second. Okay, so we'll get it started, and then as soon as it begins, you'll see it count up. In the corner. All right, thank you. Let's see.
Do I need to like press like the visitor view or something?
There we go.
Hello, everyone. My name is Daniel Flores. This is my summer work. It's not robots or AI. It's actually about bees and fruits. So we're going to get to talking about my summer project with the NSF and the Missouri Botanical Garden, investigating pollinator diversity and how they affect their fruiting crops in the family Rosaceae. So that's apples, pears, peaches, cherries. So this is a little bit about my story. I'm from Texas originally, and then I moved to with my family to Radford, Virginia, where I was then looking for a school to go to. And so I went down I-81 and across the mountains, and I found myself here in the beautiful state of Kentucky, and currently attending undergraduate biology here at UPIKE. So let's talk a little bit about the trees here. So the focal species for my study was orchard trees, like I mentioned, in the family Rosaceae, cherries, peaches, pears, apples, things like this. It's kind of really important to mention that all of these trees require cross-pollination. So they need an insect to mediate the pollination, crossing one pollen from one individual flower to that of another. They can't pollinate themselves. And the level of this pollination directly affects the fruit and quality in these trees, such as like appearance, size, mass, things of that nature. And so here's what my favorite part is, the bees and the crawlies, as I call them. And so we've got butterflies here, we've got ants here, and like I mentioned, the rose family absolutely needs insects or animals to carry pollens across their flowers to ensure that they're getting a thorough pollination and increasing yields in these orchards. And specifically, I wanted to look at how the abundance of insects, so the sheer number of insects, and the richness or diversity of insects affected these trees. And this is important because unique behaviors from these different organisms result in different strategies to pollinate, which in turn creates a more effective pollination across these species of trees. And so what we kind of know, like I mentioned, cross-pollination is vital. The fruits aren't viable, they aren't pretty, and they're smaller and kind of deformed, and that's important in the market of fruit. That accounts for $361 billion annually in fruit crops. So it's very important that we have good-looking fruits, otherwise they don't get bought. Pollination behavior, such as flower fidelity and their strategy for achieving pollen or nectar for that case, really impacts how successful the flower is pollinated and in turn affects fruiting quality. So this study was conducted, as I mentioned, through the Missouri Botanical Garden, which is located in St. Louis. And so St. Louis alone has 200 species of just bees. And bees aren't the only insects that are visiting these flowers. Beetles and all kinds of other things, butterflies, are also visiting these flowers. So there's a really great benefit for promoting this idea of ecological redundancy, or many different species visiting a flower to achieve pollination. And previous studies have showed that native bees specifically, in Osmia bombus genus, so our burrowing bees, our bumblebees, are much more effective pollinators than that of honeybees. They nearly double every pollination statistic other than pure abundance than honeybees do. Honeybees are there in more numerous amounts, but in levels of spreading pollens carried, trees visited, and things like that. All of these native bees are more effective. Not only that, but honeybees are less effective pollinators, especially because of their behavior. They have this unique behavior of standing on the flower petals and accessing the nectaries from the side of the flower, so they don't spread pollens across the stigma of the flower, which is important. So traditionally, orchard studies are done in a more traditional setting, just like open air, rural settings. And so we really wanted to focus on St. Louis urban setting. And here's just a map, a heat map of urbanization and impervious surfaces throughout the St. Louis area. So parts that are really bright pink are very covered, and then the darker pink they get, the more buildings and stuff you see. So big expanses of pink are concrete and roadways, whereas the more dark kind of reddish color is buildings and things like that. So this kind of is testing the habitat intensification theory. So the more intense a habitat is for these soils, negatively impacts the population in those, because 80% of our native bees are ground nesting, so they need soft soil or a suitable substrate to be able to establish a habitat and pollinate our flowers. Our native bees also come out earlier and fly better and pollinate better in suboptimal conditions. So windier and colder days, we see more native bees, which is important for rose family because they bloom kind of early in the year. Like right now, apples are starting to get going, and these plants are needing pollination regardless of the rainy, cold weather. Community orchards were kind of a staple in this study because of food scarcity in urban areas. So they can benefit from these kind of sustainable practices without much upfront cost just by promoting habitat. So really what we wanted to discover was how does these statistics of diversity and abundance, so number of species and then number just in general, increase the fruiting quality in these trees. And so we studied 16 gardens across the greater St. Louis area, and they range from suburban to exurban. Suburban is like neighborhoods, urban is very active, you know, a lot of businesses, a lot of nightlife and stuff like that happening in the area. And exurban are more abandoned lots, still a lot of impervious soils and things like that. And we focused specifically on apples, pears, peaches, and cherries. And so to collect data, we went out here. You can see my good friend Jack there in the second picture. He's counting apples. He's the only one that can do this because he's the first one that started doing it, so he's the counter. And he counted 1 meter squared in all of the cardinal directions. And then we also measured length, width, circumference, and mass. And so as far as the pollinators go, they netted pollinators for 5 minutes per tree during favorable weather, so warm, sunny, non-windy days were favorable conditions. And over 2 years, that lab in St. Louis at Webster University had collected over 1,000 insects. I helped in pinning and identifying and cleaning these insects. And as you can see here, it's just a collection of all the different insects there. You can see some beetles, some bees, some flies. So where we really happened to fruit mass, we didn't see any kind of significant change in fruit mass in either of the measures of abundance or richness. Neither did we that for length. We didn't see any kind of significant difference. We didn't see anything for circumference either, so not really worth mentioning other than that we didn't find anything. But we did, however, find something that is contradictory to most traditional literature, that the higher abundance levels, we saw less wide fruit, so the width decreased as the average abundance for an orchard increased, which is very interesting. But we didn't see the same correlation with species richness or that diversity measure, the amount of different species. So we did see that as abundance increased, the abundance increased, the number of fruits on a tree were increasing as well, which coincides with what we've found in traditional orchard systems. But you can see we found a non-significant finding that richness actually had a decrease in numbers of trees, which could be an interesting thing to tease apart. So just kind of discussing a little bit about what we found. So the fruit yield and pollinator abundantly did significantly affect these measures. It decreased width, but pollinator abundance increased counts. So that's kind of interesting because, you know, like I said, most studies say the opposite, that as increased diversity happens, increased pollination happens, and so abundance and all of these other measures also increase typically. A few limitations to our study was the identifying insects. A lot of these insects are very small and hard to identify, and we were working through them as quickly as we could. And at the time of this study, which was last summer, we'd only identified them to genus, which is a good capture, but not the best capture of diversity. So kind of hindsight or results, you know, thinking about urbanization and pollination, habitat reduction and increased impervious soil structure is detrimental to our ground nesting bees, and we can't piggyback on the idea of honeybees pollinating our crops. Further analysis needs to be done on the urbanization, so someone in that lab is currently working on impervious soils and pollen fidelity, so how loyal they are to a flower or their preferences based on that measure, and then I would like to coincide that data with the data I've had. And so most studies say pollinator abundance positively affects fruit yield, which is what we saw. So maybe we should be promoting, instead of external or supplemental colonies or bees coming into an orchard, so like a rent-to-bee service, rather we could try to do some kind of management to knock back that habitat intensification and just attract bees naturally and leverage our diversity and our native bees over the non-native honeybee. Thank you. These are all my, I'd like to give a special thanks to UPIKE and everyone that's involved in coordinating this great event. Thank you. Any questions on bees? Thank you. So we found a great deal more bees than we did any other insect. The second most common insect was actually the flower flies. They tend to like the nectar, but bees by far were the most dominant species. There's just a lot of bees in St. Louis. They actually are one of the most diverse cities in America. Thank you. Yeah, you're welcome. Thank you all.
Thank you very much, Daniel. All right, so our last student presentation today will be Colton Hall from the College of Art and Science. We'll get this started here, and then as soon as we start the presentation, what you'll see is in the corner, they'll actually have it count up, so you can keep track of your time.
Okay, so whatever it is. Okay, thank you.
All right, there we go.
All righty then, it's great to see y'all here today. And, well, I'll be presenting on minority and folk religion in our very own Appalachia. We'll be covering everything from branches of Christianity you only see in this part of the world, and even the rise of LGBT, witchcraft, and paganism. We've met everyone, to say the least. But first, let us start with somebody you may have heard of. You probably know somebody who follows one of these churches, the Old Regular Baptist Church. These are churches with a very peculiar theology. They are Calvinists. They only believe that adults should be baptized. But they also are very peculiar in how they live their lives. I remember hearing my mother, when the Old Regular Baptist Church was more popular at this, yes, it still exists today, you could tell if somebody at a school was an Old Regular Baptist. The women do not cut their hair. The men, even when they go to swim, will wear pants as opposed to shorts. Very conservative lifestyle, one might say. And it's a style of worship you don't always see in other areas. Sometimes multiple men, not just one priest like we would get at the local Catholic church or like Paul Potter does at Cornerstone. Multiple men play the role of preacher. Now, we also visited a snake handling church. I hate that I have to disappoint the audience here and tell y'all that we did not get to see them handling snakes. It feels as if most people who handle snakes were born into a church that handles snakes. Snake handling is technically illegal, but these churches still do it. They have a very literal interpretation of a certain verse in the Bible, meaning they will literally be able to triumph over snakes. They don't see the snake as a metaphorical snake that we see, for example, Catholic art of Mary stomping on a snake. No. And the belief is their faith in God will prevent the snake from biting them. These were very long worship services. They did services every evening of the week except for one. The first hour was nothing but singing, and some men would get so ecstatic they'd run around in a circle around the whole church. Women would lift up their hands and start crying, and the man running around would bless them. You've never seen anything like it. Now, this is actually what inspired the project, the folk healer. Ask any grandparent who lives in this area, and they will tell you that for the longest time, Appalachia didn't have the greatest medical care. You also probably know this, we sadly still struggle with this. Appalachia has been riddled with poverty at times. So what do? You can't afford to go to a doctor, or maybe you straight up can't go to a doctor. I remember my grandfather taking people all the way over to Ashland just to go to the doctor. Well, it's time to consult the folk healer. For many older ladies would do it. This right here is where we have the concept of some call them granny witches, but that's a modern term. Nobody back then would have called themselves a granny witch, even if you want to call what they do folk magic. You can see right here a book, Long Lost Friend. I actually have a copy of it with me. This is a book full of folk remedies. If somebody is bleeding, for example, there's a prayer you can say over somebody. It involves one passing the hands over somebody 3 times. Then they would make the sign of the cross in a very peculiar manner 3 times too. This was very much rooted in Christian faith. The idea was that the person who was performing it was channeling the Holy Spirit, and that the person who was receiving it was being healed by their faith in God. I guess a lot more involved version of a tent healer. And books like Long Lost Friend were sold at gas stations. This was pretty common in mail order catalogs, from what I know, too. However, it wasn't always that. Some people would create little charms they would carry around on paper. Some people would use herbalism, for example. A lot of the older folks you might know in the area have likely taken medications that were homemade, made from herbs. Sadly, a lot of people do not practice this anymore. Sometimes there's the belief that if you die with the ability to talk out fire, for example, if somebody was burnt, they would speak over the burn and the wound would fly, supposedly leave. Well, you had to pass that on to somebody, and from what I know, the person, at least the holler my family comes from, died without giving it to somebody. Or you would have very specific circumstances that don't really exist anymore. For example, thrush, a fungal infection of the throat, was supposedly cured by some mixture of the number 7, which goes all the way back to European folklore. I remember every holler had some remix of something involving the number 7, the 7th son of a 7th son, or a 7th son who never met their father. Now, we went and interviewed prison religions twice. I think prison religion is a very good thing because I know people personally who developed a religious practice in prison, and they did not commit a crime again. We interviewed prisoners at the Wheelwright prison, and I can promise y'all right now, every religion you can think of exists in that prison. Mormons, Islam. We even saw Norse pagans as one group we interviewed. We interviewed Norse pagans and Messianic Jews. And what you will find is that these people take their faith seriously. This is one of their favorite things to do. One of the few times in prison that they are not meant to be suffering. Maybe it doesn't mean every inmate has to become religious, but maybe we could learn from that. Maybe we could learn that a positive attitude and could maybe change our lives a bit. And now we have minority religions. And what we mean by mainstream religion, first of all, let's clarify this. Well, Christianity, obviously, but the churches we interviewed, snake handlers and irregular Baptists, are not mainstream Christian denominations. When we say mainstream Christian denomination, let's think of stuff like the Catholic Church, the Methodist Church, and the very big one here, Southern Baptist Church. You will notice a lot of the people, Christian or not, had a problem with the current Christian zeitgeist we see in the area. And the one denomination most people were pointing fingers at was the Southern Baptist Convention, which is a very conservative Christian denomination, or at least compared to some of the mainline Protestants like the United Church of Christ or Episcopal Church. I personally have family who have become Buddhists. Some people really like the Buddhist philosophy. In fact, I see a lot of Christians even realizing that Buddha and Jesus had a few points in common. We interviewed also a lady who had converted to Judaism. This was pretty unique because Judaism is not what we call a missionary religion. In Judaism, a Gentile, what they call somebody that's not Jewish, is not obliged to become a Jew. Jews do not have the concept of heaven and hell like we see in a lot of Christian denominations. No. So it surprised me that a lady converted to Judaism in the area, because not only is converting to Judaism a very hard task at times, the rabbi will deny you thrice before he will finally give you permission to become a Jew. And even then, the whole process takes at least a year, and I know it's a lot more intensive than RCIA, the year-long process to become a Catholic. So I was very surprised, and what I've noticed is anybody who converts to any other religion, or is part of a minority religion at least, convert or not, tend to be very devout in their faith. The old regular Baptist preacher we interviewed, Jeff Little, he said, direct quote from him right here, "God led me here." These people feel a deep connection with their faith. A lot of these, as we are about to see, are even people who have suffered, not just prisoners, but another kind of zeitgeist people struggle with in this area. So, and you'll find that people who practice minority religions outside of big cities like Huntington and Charleston, oftentimes are solitary practitioners. The Jewish lady we met, I don't know for sure if she had fully converted to Judaism because she had to attend an online synagogue. We don't have a synagogue for miles. But the point is that they exist and they're here. Shelby Valley of the Pagans, if you get that reference, you're awesome. But anyways, there's been a recent rise of paganism. Now, I'm not here to say those filthy pagans or anything like that. What I mean is instead the rise of nature-based spiritualities or attempts to revive European paganism. For example, we saw Norse pagans at a prison. These were people worshiping gods that were worshipped thousands of years ago. Thor, Odin, Freya, to name a few you've probably heard of. We also talked to Celtic pagans. I sadly didn't catch what deity she worshipped, though. We also interviewed two witches. Now, witchcraft is not a religion. There are religions based on witchcraft, such as Wicca, but to be a witch is not to be religious. I've seen, if you can believe it, atheist witches before. And what you will find is the old regular Baptist church and snake handling churches we visited are very conservative churches. However, a lot of the pagans and witches are very progressive. In fact, you'll find LGBT people tend to be more willing to embrace this type of stuff. Of course, not every LGBT person is like that, but a lot of the people we interviewed were LGBT. We were much more likely to meet somebody who would use something like they, them, here as opposed to at an old regular Baptist church, to say the least. And a lot of people, since Appalachia is a more conservative region, a lot of LGBT people are able to find community in groups such as witch convents. Now, this does lead to some controversy in the area. What I've noticed, and so did Raw Music, who worked with me on this project, it's going to happen very soon. That garage, quite a few of you are probably parked at as we speak. They, during Hillbilly Days, do this thing where they have merchants in there, vendors. We're getting close to our time. Can you finish in about a minute? Yeah. Vendors. And these are people who will sell stuff like crystal, statues of goddesses. How is that selling in the Bible Belt? There are 5 full full stands of these people in the Bible Belt, all well populated. And this has led to concerns over gentrification, for example. Some of the folk practitioners I met accused these people of being from gentrification, richer, usually white people moving into the area and bringing these newer practices instead of anything native to Appalachia. What do you think? I'm going to leave that one up to my audience. The end. All images created, Creative Commons, thanks to Raw Music and everyone interviewed, and of course, you, because you listened to me.
Thank you very much, Colton. We appreciate that. If you have any questions for Colton, please write them after the session. All right, and that's the end of our student oral presentations.